years ago. For the last 4.5 billion years, the most important factor in breaking bedrock down into sense that clay has been bacteria and fungi. They are what do that breakdown. So, as long as we're getting bedrock converted into calcium clay and up with that organisms in the soil, do you ever have to worry about phosphorus or sulfur or magnesium? Would you ever have to worry about it? No. How many of you have ever went out and found something in your soil? Little turtle, aha. Not, have you? Or you wouldn't do your own plants. What if you have pure organic matter? What if your soil was pure organic matter? What are the nutrients in organic matter? In that plant leaf? Any calcium in that plant leaf? Any sulfur? Phosphorus? And aren't they in the right balance to grow other plants? So if you have a soil that's mostly organic matter, do you have the nutrients to grow plants? Absolutely. But are they in a soluble form? No. We got to convert them out of plant material from the organic, and we got to convert them back into what form of nutrients? The soluble ones. And who's going to do that for you? The organism. So, organic matter, another very important component of soil. It ain't soil unless you've got organic matter. And, of course, then, organisms. It all revolves around this. Which organisms are present? Which organisms are functioning? You have an adequate cycling of nutrients, so you're always producing enough soluble material so your plant will never be limited in the nutrients available to it. And it is the biology in that food web that we have to understand. So, here we are, cane growers. We came to Hawaii, and we started plowing the soil so we could put our cane plants in. We could start growing them. As soon as we harvested, how do you get rid of that thick layer of organic matter that's now sitting on the surface of the soil where a whole bunch of diseases are growing in that material? So what do you do with that thick layer of organic matter? Burn it. Duh. What a stupid thing to do. Because you're just blowing off into the atmosphere all of the foods that feed the organisms that convert into that soluble form. Because we don't have enough patience, because we don't understand what we're doing to the soil when we till and kill off those organisms, when we put on inorganic fertilizers and kill those Do inorganic fertilizers kill biology in the soil? Oh yeah. Every single inorganic fertilizer is a salt. Anytime you're putting on more than 100 pounds per acre of an inorganic fertilizer, you're killing beneficial organisms in your soil. Where are the data? Well, you can't look at the experiments that the chemical fertilizer companies are doing. Because, gosh, why would they want to bias the results and lead you astray that chemical fertilizers don't kill anything important in the soil? Why would they want to do that? Convince you that there's no harm in using those inorganic fertilizers. So, if you're already starting out with a life with no soil, yes, you have to use inorganic fertilizers. As soon as you get the biology in the soil, you don't want to be using those inorganic fertilizers at all, ever again. Because anytime you put more than 100 pounds per acre, you're killing off that very life that does the nutrient cycling for you. Your choice. I don't really care which way do you want to do it, but ultimately, where do human beings have to go? They have to get off the chemical fertilizer. Should we call that a chemical dependency? Don't we have to get off that little trip in order to be able to maintain soil? Yes. So we have to understand this. Every inorganic fertilizer is a salt. If you're putting out lime, what's the recommendation typical on a soil chemistry report? What's the recommendation that you get from a soil chemistry lab for the amount of lime that you have to put on. Less than 100 pounds per acre. A ton per acre. I've seen soil chemistry reports that recommend 8 tons per acre. I've seen them recommend 12, depending on the pH of your soil. What causes pH in soil? Yeah, see, most people don't really understand that. What controls pH in the soil? Well, oh surprise, it's biology. You got the wrong biology, you're going to have really acidic soil. You got the right biology, well, then it depends. What plant are you trying to grow? So, see where we got a little bit, I want to explain all of this to you. Yep, got to get there. So, understanding the biology. Pesticides. Is there any pesticide that only kills the target organism? All of them kill non-target organisms. You use a pesticide, what have you killed? Wouldn't it be useful to have a simple testing method to be able to tell you what you just killed? You had to save your plants. You had a mildew outbreak, and it was wiping out. You have a Phytophthora outbreak. You have black Pseudotsuga killing all your bananas. You've got some fungal disease that's just wiping you out. Okay, use the pesticide. But then, you don't ever want to have that disease again. So how do you get this system into a sustainable condition? We've got to have the biology. If you don't have the proper biology in your soil and on your leaf surface, you are stuck with having to use those, oh, they're real inexpensive, aren't they? Those pesticides. Yeah, kind of ouch when you have to use them. Use them if you have to, but you shouldn't have to. So how do we get you out of that need to use those toxic chemicals? And if you've used a toxic chemical, you're going to have to get the biology back in there. Because what else did you kill rather than the disease?
When you were speaking about liming, that includes like calcium carbonate and dolomite in excess of the calcium.
Yeah, so lime is calcium carbonate. Dolomite is a slightly different compound. It does contain calcium, but it actually contains a great deal of manganese. So calcium-magnesium ratio, isn't that a way we're putting calcium on, is to get that calcium-magnesium ratio right? So if you put on dolomite, you're doing that. You're going to have more magnesium being made available than you are calcium. So you're making things worse. You're tying up your calcium with that excess magnesium that you're putting on when you use dolomite. Why would some company want you to be out there putting on dolomite? Because what would they like to sell you next year? More. See, more on farmers. So you have to learn chemistry. So if you really want to understand what's going on, you do want to know some of the tricks that have been put over on us. I have had growers that have come into me, and they'll bring in their whole stack of soil chemistry reports, and they're just angry as can be. So a grower just outside of Davis, California, comes into my office, and he hears this stack of chemistry reports, slams them down on the desk in front of me. I have been putting out 3 to 4 tons of lime every year for the last 32 years. I did my soil chemistry test this year, and guess what? They recommended another 3 tons of lime and half a ton of gypsum to go out. You know what? It's not working. What do you think when somebody does that? Slow learner, aren't they? 32 years. And I finally figured out that putting on that much lime is not going to do much for you. How do you solve the problem? How do you fix this permanently? Biology. You have to get the organisms that are going to solubilize that calcium out of the sand-filled clay, out of your rocks, out of your organic matter. Tie it up in the bacteria and fungi. Hold on to it so it doesn't leach. It doesn't wash away when you get water moving through your soil. Do you have a little water moving through your soil here?
A little, yeah.
Just a little, on occasion, now and again. Like it was pouring rain when I flew in. So yeah, you have a little water movement. We gotta hold it, keep it in the soil so it doesn't wash out. Keep your nutrients where they're supposed to be. Then we have to have protozoa, nematodes, good guy nematodes, microarthropods, come along and eat, earthworms come along and eat those bacteria and fungi and release those nutrients in a soluble form. Now, do we want soluble nutrients being made in our soils anywhere far away from our root system? No, because we're going to lose them. Wash that water, they move soluble. That means they're dissolved in the water. And if you got water moving through your cell, where are your soluble nutrients going? They're down there in your groundwater. They've moved beyond your root system, perhaps. So we want our roots growing as deep as they possibly can into your soil. We want most of this nutrient cycling going on right around the root system. How does Mother Nature manage to do this? Well, where's most of the food for your bacteria and fungi being released in the soil? Where's most of that food coming into your soil system? Surface is putting down dead plant material, but that is not the place where most of the organic material is actually being released into your soil. Sorry? A lot of it, but not most of it. It's from the plant itself. When plants grow into your soil, that root system, as those roots grow, all of those roots release exudates. And that is the food that bacteria and fungi grow on. When we're looking at soil around that root system, we are looking at 1 million bacteria per gram of soil around the root. We have 10 to the 12 bacteria per teaspoon. 1 million bacteria. We are holding boatloads of nutrients right there next to the root system. And the plant is in control of who that is that's growing. Who is which bacterial species? We've got miles of fungal hyphae growing in that zone right around that root system. Those bacteria and fungi are immobilizing and holding nutrients right there in the root zone, so those nutrients never move away from your plant. Ignore the person coming in the door. So we're holding our nutrients right there in the root zone. What else do those bacteria and fungi do for you? They are going to compete with, inhibit, and consume disease-causing organisms, pests that might attack your root system. That castle wall that we're growing around that root system is going to protect your plant from fungal diseases, bacterial diseases, nematodes, all of those problem organisms protecting that root system. As long as we've got the bacteria and fungi in that soil, so as your root grows through and releases those exudates, we're going to be growing a castle wall around that root system. We're going to protect that root system. We're going to hold our nutrients right there.
You can define exudate for me?
Exudate. What is an exudate? That's one of those wonderful scientific words that we use to try to confuse you guys. Yeah, I know more than you do. Isn't that what we do that for? It's our own little language. So what is an exudate? It is mostly sugar, a little bit of protein, and a little bit of carbohydrate. So mixed. Again, what kind of sugar? Well, it depends on what the plant needs. What kind of sugar is it about? What kind of protein? What kind of carbohydrates? There's some really interesting work that's been done by Joyce Loper, L-O-P-E-R, where she has looked at strawberries and what are exactly the exudates coming out of that strawberry root system. And that strawberry, this first centimeter of root is putting out a different set of exudates, and the next centimeter puts out a different one, and the next micrometer puts out different. And as you go back along that root, there's all kinds of different sugars, proteins, and carbohydrates being produced. Why would your plant do that? Well, here I am talking to you about sugars, proteins, and carbohydrates. If I asked you to go into your kitchen and make a mix of mostly sugar, some protein, and some carbohydrate, what am I asking you to make? So let's think about it in my kitchen. Here's my bowl. I'm going to go get sugar. I'm like, what's sugar in your kitchen? White sugar, brown sugar, honey, molasses. Because think of all the different kinds of sugars that you could put. So if you put mostly molasses in there, or you put mostly brown sugar, will you be making a different kind of cookie? A different kind of cake? So what is the mixture? How much of which kind of sugar? And you're going to be making different things to grow different organisms. Okay, so mostly sugar. Now protein. What's protein in your kitchen? Yeah, throw a couple eggs in there. Maybe some milk. Different kinds of protein. Mix that in. Now carbohydrate. What's carbohydrate in your kitchen? Flour. So put that in. What is this a recipe for? Cakes and cookies. Your plant is dumping cakes and cookies out of the root system. And then it's doing that in order to grow precisely which species of bacteria, which species of fungus is needed to benefit that plant to the greatest amount. This side of the root system lacks iron. So up here on my leaves, my flowers say, hey, guys, down there in the soil, I don't have enough iron up here. Iron's needed. So the chemical message goes down to that root system. Put out the exudate to grow those iron-solubilizing bacteria, iron-solubilizing fungus. So on that sand-filled clay, or in that mineral material right around your roots, we're going to solubilize. We're going to take the iron that the plant needs, gets pulled into the root system. We're still missing one step, though, aren't we? Bacteria and fungi solubilize and hold it in their body. But that's not available to your plant, is it? Because all you got growing in your root system is bacteria and fungi. Your plant is out of luck. Not going to be able to get the nutrients because the plant has to have soluble forms of iron. I better get a chelated iron going, being released. Oh, there's another good word. What's chelated mean? So when we're looking at iron, or potassium, or sodium, or calcium, what's the form of calcium your plant takes up? It's not calcium carbonate. What form of calcium does your plant take up? It's chelated calcium. How do you get your calcium chelated? What's the step to get those nutrients chelated? Now, what does that word mean? I'm kind of bouncing around in my presentation. Hopefully it makes some sense, though. So what's chelated? Here's how you spell it. C-H-E-L-A-T-E-D. Chelated. Wonderful chemical term. Most people have no idea what that actually means. Chelated just means that calcium molecule is attached to an amino acid. That's all that means. Where did amino acids come from? They're inside the bacteria and fungi. So when the bacteria and fungi take up any mineral nutrients, the way it's soluble inside the bacteria or the fungus is as a chelated form. Now, how do we get these nutrients out of bacteria and fungi and released in those soluble forms? Protozoa have to eat the bacteria, or bacterial-feeding nematodes have to eat the bacteria. When a nematode travels down on the bacterium, there's way too much nitrogen, too much phosphorus, too much calcium, too much magnesium. Every mineral nutrient is in way too high concentration inside that bacterial body. And if the nematode or the protozoan doesn't poop it out, it's going to kill that nematode. It'll kill the protozoan. So our predator, the protozoa, the nematodes, the microarthropods, our earthworms, poop out all of those forms as chelated. They're going to poop out the excess calcium, calcium, phosphorus, those sorts of things in a plant-available form. I'll talk a little bit about some of the special cases. We'll talk about nitrogen. We're going to talk about phosphorus. We can talk about sulfur. Some of these things, slightly different. They don't come in chelated forms. Can I go off on this tangent? Anyone going to mind if I go this way? What are the soluble forms of nitrogen? What are the forms of nitrogen that your plant takes up? Yeah, so the two soluble, can you see this when I'm writing this up here? Can we turn the lights on for just a second? We may have to have lights on, lights off. Very low. afternoon. So, ooh, look at that, you can see it now. Nitrate, one of the soluble forms of nutrients that plants take up, but do all plants take up nitrate? No. Nitrate is actually detrimental to a whole bunch of plants. If you're trying to grow a perennial plant, don't be putting nitrate out because you're going to cause disease. When you're looking at trees, when you're looking at perennial shrubs, you will cause disease on the root systems if you put nitrate into the soil. There's been 2 books written on this in the scientific literature. I don't have to go over this. It's not my work. It's been known for a very long time in science. NH4, which is properly called ammonium, not ammonia, although most soil chemistry reports will make a mistake. They will call this ammonia. And it's not. What is ammonia? It's NH3. And this is a gas. If you've got ammonia, is it going to stay in your soil? No. Say goodbye to it. It's going to be lost as a gas. So, not a form of nitrogen that your plant can take up. So, we don't ever want to produce ammonia in our soils. What's the only way to make ammonia? It is an anaerobic product. It is only produced under anaerobic conditions. So you go out to your nice puddles out there in on your soil, and you start going, ooh, I smell ammonia. What's that telling you about the nitrogen content in your soil? It's going away. You no longer have nitrogen left in your soil to grow your plants. So we don't want to have that. You go out to a compost pile, and you smell the ammonia coming up. Sorry, it's not compost. Because as soon as we go anaerobic, the only way to produce ammonia is if you went anaerobic. So in that material that you might have once called compost, you start smelling ammonia, you cannot call it compost anymore. Because compost is, by definition, aerobic. Composting is the oxidative decomposition of a mix of organic matter. You go anaerobic, it's not compost. Stop calling that garbage compost. If you smell ammonia, what do you call it? If you smell ammonia, if it's gone anaerobic, what do you call it? Garbage. It's reduced waste. Don't call it compost. Don't make that mistake. You will not get any benefit out of an anaerobic material because where's your nitrogen gone? Yeah. So not a form of nitrogen that we want. So nitrate and ammonium are the 2 soluble forms of nitrogen that different kinds of plants take up. When we're looking at what kinds of plants, when we categorize them generally, what types of plants take up nitrate? Weeds. You want to grow weeds? You like weeds? Put out some nitrate because you're selecting for that community of plants. The weedy species require high levels of nitrate. You like thistle? Make sure that your nitrate concentration gets above 50 parts per million because all your thistle seeds is going to germinate and grow. Perfect. What if you don't like thistle? Don't ever let the nitrate level get up above 50 parts per million. How many of you like nutsedge? So don't ever let your nitrate level get above 10 parts per million in your soil. So what's the recommendation coming from your fertilizer salesman? How much nitrate fertilizer are they telling you to put out? 450 pounds per acre. You know what that is in parts per million? Woohoo! Yeah, you are going to grow a bunch of weeds. So if you want to do that, fine. But if you'd really like to have something else growing, maybe you should not be doing that anymore. When we're looking at ammonia, what kinds of plants use ammonia? And these are our perennials. Now, what if you want to be growing some grass? What if you want to be growing veggies? Well, what's really important here is that you get the balance of nitrate and ammonium correct. You're growing shrubs, you're growing trees, you want almost all of your nitrogen in this form. If you want really early successional plants, weeds, you want it all in that form. But let's say we're trying to grow most of our veggies. We want a fair amount of nitrate, but we must have some ammonium, and that selects for these to grow instead of those. What if we want a good grass species, really mature? We want to grow some cows, we want to grow some horses, some llamas. So with our grasses, we need to have an equal amount of nitrate as compared to ammonium. And we're going to set the stage to grow them and not vegetables, and certainly not weeds. What is it that causes ammonium versus nitrate in our soil? What is giving us nitrate versus what gives us ammonium? Guess what? It's biology. There's a few more details that I have to get you into to understand how we cause this balance. We have to understand what bacteria do. We have to understand what fungi do in order to understand that balance, in order for you to get it right. So, do we need that little microscope so that you can get the information on what's the balance of bacteria and fungi? Because that's what determines your ratio of nitrate and ammonium. If you don't get it right, you're not going to be selecting for the plant you want to be growing. Too bacterial, you're going to get it too weedy, because bacteria are what help make nitrate. Fungi are what help make ammonium. So we have to have that balance. Okay, so there's a few interesting things that we've got to understand in all of this. And how do we release nitrate and ammonium? Yes, bacteria or fungi being eaten by protozoa, bacterial feeding nematodes, fungi being eaten by fungal feeding nematodes, or microarthropods. So you need to know how many bacterial feeders you have in your soil? How many fungal feeders? Do you have to know your microarthropods? Do you need to know whether you've got lots of protozoa or nobody? So there's a little bit of assessment. Luckily, it doesn't take much to do this assessment. Bring a soil sample in. We're going to weigh out 1 milliliter volume, put it in 9 mils of water, shake it for about 30 seconds. A drop of that goes on the microscope slide, put a cover slip on it, under the microscope, and we look at it and we go, oh no, all you've got is bacteria. What are you growing? Weeds. No protozoa, no nematodes. Oops, you're not going to grow anything, basically, because your bacteria is sucking up all your nutrients and keeping it inside the bacteria. Or we look through the microscope and we go, wow, look at everything moving around in there, and the flagellate's going this way, and it's doing this, and the protozoa, and we're just having a grand old time. And it's like, okay, whatever you want to grow, you can do it. We're just going to modify that ratio. It's that simple and it's that easy. Okay, and it takes a little experience to learn what the different critters look like. But if you want to come to Rodale for, 2-3 weeks this summer, an apprentice, we can teach you how to do that. And the more places we can get that, people understanding this information and helping interpret for others, faster we can get sustainable. So nematodes.
Lights out.
Lights out. Where are those lights again? Yeah. So there's a bacterial feeding nematode. How many of you would ever have recognized him if you were looking through your microscope? Total magnification here is 400x. This is not extremely high microscope. You don't have to have oil immersion. We don't have to have really expensive microscopes. There are microscopes for about $300, $400 with cameras. So you can start taking your own pictures. Even if you can't identify it, you can send it in to me or to somebody that I work with, and they'll help you identify it. So nematodes, cute little mouse, isn't that cute? Here you go, bacteria. whips them right down. And so that particular mouth structure with these cute little hairs on the end, bacterial feeder. This is an Orlanid. So it lives in vegetable roots. That's its favorite place to hang out. It eats aerobic. If you go anaerobic, this guy's going someplace else. Think about your own house. If you walked into your house and it stunk to high heaven, how long would you stay there? Yeah, I'm leaving. So if it's an anaerobic condition where it lives, it's gone. But aerobic place, it's going to stay. It's going to make a house. It's going to be doing what it's supposed to do. It releases nutrients from the bacteria into a plant-soluble form. And it's going to be releasing nitrogen, phosphorus, sulfur, magnesium, calcium, potassium, sodium, iron, you name it. However many nutrients your plant is looking for to take up, it's being released. So is there enough nutrient cycling going on in your soil around the roots? Not out there somewhere, around the roots. So how is your plant making sure that all the nutrient cycling it needs to have happen is happening right in that root zone? Exudate. Your plant's putting out the food to feed the bacteria and fungi. So we've got a million bacteria growing right around that root. We've got miles of fungal hyphae growing right around that root, preventing diseases. But now we're attracting the protozoa, we're attracting those good guy nematodes, we're bringing in the microarthropods, we're bringing in the earthworms, and they're chowing down on those bacteria and fungi, releasing soluble nutrients that your plant requires. So the place where most nutrient cycling is happening is right in the root zone. Isn't Mother Nature tricky? Isn't it just elegant in the simplicity of how she put it together? Just because we as human beings have never figured this out before, doesn't mean it's not happening. So back in 1950, when we started doing the chemical agriculture thing, did we know that this was what Mother Nature was doing? We had no clue. So is it our fault that we fell into that little trap? But now that we do know, now that there's lots of evidence to show that this kind of nutrient cycling is what's happening in the soil, we need to move to a more sustainable system. So this nutrient cycling system I've been talking about was first published in a paper in Ecological Monograph. So back in 1985, this ecological monograph and the references, a copy of the paper is on the Soil Food Web website. So the paper is Ingham et al. And there's heard, there's like 7 other authors on this paper because I was doing the bacteria and the fungi. Somebody else was doing the nematodes, my husband. Someone else did the microarthropods. Somebody else was helping with the soil. Somebody else was helping with the plants. Somebody else was doing the nitrogen analysis. So it was a lot of people, a whole team. So Ingham et al., I think it's 1986, it might be 85, I'm not remembering exactly, but it's an ecological monograph. The original paper was 75 pages long. And it goes through bacteria. Why are they important? What do they do? Fungi. Why are they important? What do they do? Protozoa, nematodes. And showing the effect on plant nutrition. How did we measure that? How did we figure all of this out? Now, I am in fact not the first author on this paper. That Ingham is Ingham R.E. I am Ingham E.R. Do you think this confuses people every once in a while? And I've had people say, you're lying to me. You're claiming that you published this paper in Ecological Monograph, and you didn't. Never said I was the first author on this. My husband. I'm the third author on this paper. So, you got to go to position number 3. John is the second author, I'm the third author. So I recommend this paper for all of you who would like to read this because it's a great way to put yourself to sleep at night. It is dense science. It's really hard to read, but here's the first documentation of the nutrient cycling system. But you can see why it's 75 pages long. You can get the condensed version without all the advertisements on the various pages. Pared down to 25, you can get a copy of that now if you so care, to have a really good put yourself to sleep article for nighttime reading. When I think it's boring and I'm one of the authors on it, it's like, look out. But since this time, there have been literally hundreds of papers published in the scientific literature that repeat this work in various smaller components. Now, quite often something that just talks about bacteria and how they immobilize, fungi, how they suck things up and hold it, protozoa, and how much nutrients being released, nematodes, what is the nutrient release level in different soils under different conditions for different plants. You think temperature might affect this? Yeah, bacteria grow faster when it's warmer. They slow down when it's colder. Yeah, but if you don't have any food for your bacteria, I don't care how warm it is, they ain't growing. But you got lots and lots of food. I don't care if it's freezing. You got bacteria that will grow under those conditions as long as they have food. So it's not just temperature. How about moisture? Will your bacteria be growing at 2% moisture? Guess what? There are bacteria that grow at 2% moisture just fine. But you have to have those species there. Where do you find those species? In deserts. So if you go to the dry side of the island, you could pick up some of those bacteria that grow when it's only 2% moisture in your soil. So if you go through dry periods, well, why not go get some? And bring them over and get them into your soil. Well, how about bacteria that grow at 70, 80% moisture? Oh, there are bacteria that grow at that percent moisture. But how fast are they going to use up all the oxygen? And it's going to go anaerobic in your soil. Oxygen does not move through water very fast at all. So if you've got any kind of microbial growth in your soil when it's waterlogged, you're going anaerobic. And as soon as you go anaerobic, what is the most dangerous thing for your plants? The material that will solubilize any plant material. And it's only produced under anaerobic conditions. Alcohol. If you go anaerobic, and you have either bacteria or fungi that can grow in anaerobic conditions, reduce oxygen, as they grow under anaerobic conditions, they're going to make alcohol. So what's that going to do to your plant? Dead plant. So you drink alcohol all the time, right? Why doesn't it kill you? Oh, wait a minute, it will. You drink too much of it, it will kill you. So what's the mechanism human beings have that allow us to drink some alcohol up to a point? Yeah, it's called your liver. Now, we have a mechanism for dealing with anaerobic material. Plants, terrestrial plants, do they? No. Wetland plants, do they have a way to deal with anaerobic byproducts? No, they're called parenchyma. They are pumping oxygen down into that root system, so it doesn't go anaerobic around their roots. But if it goes anaerobic long enough, can even that ability to pump oxygen down there, can that be overwhelmed? Oh, yeah. So when did you cross that? Taro. Why can we grow taro in waterlogged soil? Guess what taro has? Has mechanisms for dealing with that anaerobic condition up to a certain point. Don't go over it, or you got dead taro. So what's the right biology? Right. Any of our wetland plants, any of our riparian plants have mechanisms for dealing with anaerobic conditions, but terrestrial plants do not. So your onions, your corn, your tomatoes, potatoes, any of our terrestrial plants, bananas, have no mechanisms for dealing with anaerobic conditions, the roots will die. It will kill them. So we cannot let things go anaerobic. You start getting too wet for too long with any kind of microbial growth in there, look out. We're going to have some dead roots. Question?
With taro, I grow taro wetland. One of the methods that we use to increase oxygen is have the water fall into the patch. But increasing the oxygen in enough water is really going to be beneficial. You don't want to stagnant water.
Yep, that's why you can't. That's why taro can't deal with stagnant conditions for very long at all. Because you start producing that alcohol, you get to a certain concentration of anaerobic alcohol conditions, and your taro will succumb.
So anything to increase the oxygen in the water.
Yep. Don't get to that point where we're harming the plants. And when you start thinking about pathogens, when we start thinking about all the bacteria and fungi that will attack and destroy your root system, the one factor that's the same with all of those disease-causing bacteria and fungi is they require anaerobic conditions in order to grow. So alcohol production, anaerobic conditions, we are selecting for pathogens, diseases. Phytophthora requires reduced oxygen conditions in order to outcompete any of the good guy organisms. Pythium requires reduced oxygen. So any of our disease-causing bacteria or fungi, you go anaerobic and you set the stage for something attacking your root system and killing your plants. So here's two mechanisms of wiping your root system out, both of which require reduced oxygen conditions in order to have the problem start to develop. Well, you go anaerobic, and are you going to get normal nutrient cycling anymore? Because protozoa are strict aerobes. The good guy nematodes, strict aerobes. What are the nematodes that require reduced oxygen conditions? Root feeders. So the root-feeding nematodes require reduced oxygen conditions. What happens when your soil is compacted? Oxygen cannot move into your soil because the spaces between the particles are too collapsed. There is no space for oxygen to move in there. You're going to go anaerobic. Now you're going to have root-feeding nematode problem. Why won't your roots grow into compacted soil? Because it's anaerobic. Not because it's compacted, but because it's anaerobic. That's what the real problem is. So when you've got water sitting on the surface of your soil, you've got a puddle. What's Mother Nature telling you? You've got compaction. And you are not getting oxygen movement into that soil. You're going anaerobic sometime soon, real soon, especially in Hawaii. Do you have a few bacteria in your soil? Probably. They're sucking up the oxygen. You're going to go anaerobic. Now what kind of plants you growing? So you're killing off the good guys. When we take a look, so here's some soil that we took 1 teaspoon of this, mixed it with about 9 teaspoons of water. So we've got to spread the soil apart out a little bit so we can see the organisms in this material. Spread it out so we can see these critters. So now we're looking at a root here. And look at the fact that the root is being attacked. See where we got the root cells, and all of a sudden we got a divot. Something's been chewing on it. Something has been eating that root system. We have some necrosis started to happen. See the dark black color along that root system? This is a disease, sick root. It's gone anaerobic. And I can guarantee to you that this is an anaerobic condition because this organism right here tells me beyond a shadow of a doubt this is anaerobic material. This is a cilium. There are 3 groups of protozoa. There are flagellates, there are amoebae, and there are cilium. Flagellates and amoebae are strict aerobes. When we deal with soil conditions, always strict aerobes, they will only exist in large numbers in aerobic conditions. But as we start to lose oxygen, the flagellates and the amoebae go to sleep. They go into dormant stages. And then the ciliates bloom. It's a high number, so we'll see lots of ciliates. How can I tell this is a ciliate? We're looking for morphology. See these short little hairs? They're short relative to the body size. Here's some individual bacteria, a little out of focus. There's another single bacterium. There's another one. Another one. We have an aggregate here. We've got bacteria growing on the silts and the sand and the clay particles. We got a little bit of organic matter in here. So the bacteria are gluing that all together. Aggregates built, the smallest size aggregate built by bacteria, beginning to form structure. But there was enough microbial growth, enough anaerobic oxygen being taken up, anaerobic conditions. Ciliates got to really high numbers, and that tells you it's anaerobic, and pathogens starting to attack the root system. They all go together, hand in hand. You start seeing a whole bunch of ciliates, first thing you got to do is get oxygen back into the system. What if this is compost? How do you get oxygen back into your compost pile? Turn it. Yeah, pretty simple, huh? Okay, maybe. Got to be there with your shovel or your pitchfork and turn it over. Or you get a turner and turn it so we get oxygen back in there. But it's why we don't want our compost more than about 50% moisture, because as soon as you finish turning it and the bacteria and fungi are growing real fast again, you can go anaerobic pretty quickly if you can't get good diffusion into that pile. In your soil, what do we do to get oxygen back into the soil? We're going to till it. What happens when you till soil? Well, same thing when we turn a compost pile. We're going to be slicing and dicing and crushing some of the good guys. So sometimes we have to turn, sometimes we have to till to get that oxygen back into the system. But then think of the damage you did to the organism. So there's a balance. Some positive, some negative. Maybe it would be better never to allow it to go anaerobic in the first place. Don't let it get compacted in the first place. When you think about soil or compost and we disturb it, disturbance factor, it's kind of like if all of a sudden sitting in this room, a great big plow, Paul Bunyan outside, Babe the Blue Ox and Paul Bunyan with the big plow coming through this room, and how many of you would be alive after that great big plow came through the room? Would it kill some of you? Yeah. How about the rest of us? If you weren't killed, are you going to come back to this room? No way, Jose. Where, the structure has been destroyed, it has been obliterated. There's nothing left here for us to do other than maybe clean up the mess. How long is it going to be until this is a functional classroom again after that kind of disturbance? Now think about what you're doing when you class, when you till, when you disturb your soil. How much biology are you going to have to put back into that system in order to build this again? How much human effort has to go back into building this structure? Is this an aerobic structure? Is there good oxygen exchange within this room? Yeah, we put in all kinds of ventilation to make sure it stays aerobic for us. Are you doing the same thing in your soil? Are you doing the same thing in your compost pile? If we disturb it, we're doing some damage to the organism. Is it what you want? Or is it ending you up in a condition where you're going anaerobic and you're making some very plant toxic materials and you're setting the stage for diseases? Thinking about what you're doing. Now, I have a number of movies that I want to show you really quick. Pretty easily. There we go. Here's a leaf. There we go. So this is a leaf that came down to the surface of the soil. And look at all the fungal strands that are growing. So time-lapse photography. Look at how rapidly that leaf was being totally engulfed by that fungal hyphae growing on it. So there's a fungus. growing onto that leaf surface. So all of the edges are where the microorganisms are going to be growing. That's where they can get their enzymes to be doing their work. These are bacteria, full of loads of bacteria. We got a little oxygen problem here when we get bacteria growing this fast to that high concentration. Let me run that by you again because it's always kind of fun to look at it all. There's the leaf. Time-lapse photography, how rapidly does that leaf decompose? But it can only decompose because we've got the fungi there. No fungi, no leaf decomposition. Wide carbon to nitrogen ratio on leaf, dead leaf, brown leaf material. Fungus growing, as the fungus releases enzymes, it starts chewing on this wide sit-in ratio material. It releases foods for the bacteria. So only after you have the fungal attack of that leaf material will you have the foods that also allow for the bacteria to be growing on the sugars that the fungus will release. So the process of decomposition, bacteria and fungi. But then think about the fact that bacteria and fungi are sucking up all those nutrients and holding them in their biomass. No protozoa, no nematodes, no microarthropods, no earthworms. Sorry, no plants available. No plant available nutrients are being produced in that system. So we've got to be thinking of the whole system. So I want to look at then how roots grow through soil. So there's a root. Growing root hairs, we have a blue stain in that system, in that liquid, and you can see where the blue stain is being put down. So when your soluble nutrients are taken up, they move into the root, and then get accumulated in that root. So looking at a single bacterium relative to a root hair, single cell of our plant, single cell, we got those two bacteria there. Now there's the root system. You can see it's growing in this agar medium. Oxygen is limited growing into that root system. So these are not healthy roots. How do I know they're not healthy roots? Now go back and take a look at the plant. So let's go back a little bit. Here's that nice cute little bacterium. Root. You can see the movement of the cytoplasm. There's those two bacteria. So this is about 400 total mag. Another. See, how can I tell that these aren't healthy? Well, look at the leaves. Are those not stick-looking plants? This is not a healthy root system. Oxygen is limited. Here's the root tip. Sloughed root cells. So this is plant cells that are being sloughed. As roots grow through soil, they slough the roots to help them slide through a material. They're putting out exudates, and the exudates in this case are feeding bacteria. Look at the capsule wall of bacteria around that root system. So massive numbers of bacteria. Can you see that? How far out the exudates are going out and growing bacteria? So much bacteria were going anaerobic in that root system, and some bizarre things are happening. No decomposition of the sloughed roots. Why not? What decomposes the sloughed root system? Those sloughed root cells. Fungi. We got to have that fungal complex where we end up with this plant material accumulating and eventually growing to these fungi instead of good guy fungi. We need the beneficial fungi in these root systems, or we just don't have good things happening. Now, what kinds of protozoa are these? Cilia. What does that tell you about oxygen condition in that root system? Yeah, these are sick, unhappy plants. They're not growing well. So, yeah, we got some nutrient cycling in the protozoa up here, but it's not going to be healthy plant growth when that happens. So let's just run through this real quick again. See the root hairs growing, taking up food, nutrients from the solution, and they put them down inside the root. But that's not what we want to have happening with normal nutrients. They shouldn't be just stuck there. They got to be converted into something else so they help the plant grow. This is about 800 total magnification, bacteria, root hair, 400 total mag. And you can still see the bacteria, no problem. lack of oxygen diffusing into that gelatin material that the root system's growing in. When it's first growing, we do have that huge bacterial castle wall around the plant, but we're lacking fungi, so we're not getting the dead, fluff root material dealt with. Got to get some fungi into that root system. So because we're going anaerobic, well, too much bacterial growth and not enough oxygen diffusion, because gelatin's not a real good way to get oxygen into things. See that root cell fluff? Isn't that cute? So what really goes on is your root grows through the soil. So what eventually happens is huge numbers of ciliates telling us some anaerobic conditions and diseases starting to grow on that root system. So you see ciliates like this member just dig those root systems out of that gelatin and get them growing normally. So huge numbers. Now let's look at some roots growing through healthy soil. So the first thing you're probably going to notice here is look at the aggregate structure. See an aggregate, and there's another one, and there's another one, and there's and there, and there, and there, and there. And look at all the space. Look at all the air passageways and hallways that have been built in that soil. So when the root moves through there, it has no trouble pushing its way through. Lots of oxygen. This system is going to stay aerobic. Root hairs don't start to grow until well past that root tip. Now there's the root tip, got lots of exudates being produced. So think of biology being grown around that root as it grows through some nice, healthy soil. Those aggregates only occur because the bacteria make the microaggregates with the glues the bacteria make. And then the fungi come along and they bind microaggregates together with the strands of fungal material. So it's a two-step process to build that kind of aggregate structure. It's got to have bacteria. They build the microaggregates that you can't see here. And then the fungi have to bind those microaggregates together into the macroaggregates that you're seeing in this picture. And then we have to have protozoa and nematodes, microarthropods, that build the larger spaces, the larger pore structures within that soil. So soil structure is a consequence of the organisms doing their thing. You're not getting oxygen into that soil without the biology building that structure for you. So soil structure is not something magical. They just, oh, surprise, there it is. It's got to be built by the microorganisms. No bacteria, no fungi, no protozoa, no nematodes, sorry. We're not going to have oxygen moving into the soil. If your root's trying to push its way down into the soil and it hits something that's so compacted that it can't push its way through there, we're not going to have healthy plant growth. So, real life pictures. Ooh. Normally, if you come for a week-long course, I go through some of my background. It's really boring, so we'll just move on. Rodale, I've been working there less than a year, so I've been at Rodale Institute for about 9 months. The first weekend I was there, I slipped on the ice and shattered my kneecap. Good way to introduce yourself to a new bunch of people, right? Wipe yourself out on the ice. So I like Hawaii a lot. I won't ever go to the top of Mauna Loa in the middle of the winter because ice and I don't get along. But at Rodale, we've been doing comparisons of conventional agriculture with organic agriculture. So two different types of conventional systems where we've been growing corn, soybeans, and something else, I don't remember. So compared with organic production systems, so we've been doing organic tilled versus no-till, and then the conventional till versus no-till. Also a manured till versus no-till. Comparing the organic system with the conventional. 30 years worth of data. 30 years. So this is not like 1 year comparison. This is 30 years of comparison. When you average conventional production as compared to organic production, grower will end up with $45,000 in their pocket on a 300-acre farm over on average in general in the organic system versus in the conventional system, $22,000 per year. So the data that shows when we compare conventional systems with organic, the organic always wins. You have to do organic right. This is not organic by substitution. This is organic getting the biology back in the soil. So it has to be putting the organisms back into the system. People want to just go out and use rotenone as their herbicide instead of heritage or instead of Roundup. I'm sorry, you're not going to get those kinds of results. It has to be real organic, where you're putting the biology back into the soil. So we don't have the weed problems, we don't have the pests, we don't have the fungal diseases. If you're doing real organic, you are always going to end up with double the income as compared to conventional. And we've been doing it for years. So in corn, soybean, and wheat, so the data, any of you who would like to get the publication, please just email me, and I can get them to send you that publication. So let me give you my email address. It's I'm on vacation. The brain has been put on hold. So let me get my card out. Make sure I give you my right email address. Here it is. How could I forget? My name with a little dot in between at, oh surprise, Rodale Institute. .org. We are a nonprofit, so .org. Or go to our website, rodaleinstitute.org, and on our website we have a copy of that summary of the last 30 years worth of data, looking at an economic analysis, an energetic analysis, compared with yields. About equal yields of corn, soybean, and wheat in organic versus conventional systems done properly. So not that much difference in yields between the two. So it's the reduced cost of the pesticides, of the chemicals that have to go into the conventional system. And whenever we have a drought, the organic systems may exceed yields as compared to the conventional systems by as much as 2 to 10-fold. Significant reduction in yields when you're dealing with conventional systems and you've got a drought. So those years are real nice for helping those things. So some of the data. Right now we have put in genetically engineered crops, corn, soybean, into these conventional systems. So we've got genetically engineered corn and soybean in our conventional rotations. So we can do this analysis when we're doing the GMO crop material, and in the first 2 years of that trial, the cost of the genetically engineered plant material has just blown the cost of doing the conventional system as compared to the organic one. Just who in their right mind would use genetically engineered corn or soybeans, because it's so much more expensive. So actual data, stay tuned, we'll have that replicated for 4 to 5 years in another couple of years comparing those systems. Looking then at what that GMO corn and soybean are doing to the biology in the soil. Initial early data says genetically engineered corn and soybean are destroying the fungal community in these soils. We don't have the structure, we don't have the nutrient holding, we are not getting the water held in the system, in those genetically engineered systems as compared to the organic. So some actual data from field trials, and those if you're interested, please get the book.
Compared with the conventional, GMO compared to conventional, are the fungus in better shape?
In the past, where we've had the conventional without GMO as compared to the organic, the fungi have been at least holding on in the conventional system, lower as compared to the organic. But as soon as we put the GMO into those soils, fungi are gone. And now all of the benefits from that fungal component of the system is gone. So GMO plants, are they putting out exudates? Is there something about their plant material that is really detrimental to the beneficial fungi in your soil? Absolutely. So we'll have the replicated data in another year. We're planning on 5 years of the GMO part of this trial. So another couple of years before we got all 5 years. We're in year 3. So we won't have 30 years. I'm not putting up with GMO plants in my organic farm for more than 5 years. That's it. They're killing my fungi. Lots of new things. So those of you who might be interested in some of these projects and you would like to come and be at Rodale, we are doing internships. We'll give you room and board while you're there, but you got to get yourself to Rodale, and in exchange for your training, we want some work out of you during the year. So no salary, but we'll pay for room and board while you're there. So whether you want to come in April and stay until December, you want to come in May and stay until August, work it out with me. Send me a resume, send me a little letter that says, what would you like to work on? What do you want to learn while you're here? And what are the dates that you could come and work with us? So trying to get this knowledge spread all over the world. And of course, compost is one of the big things.
Take a break.
Okay, break time. It's already 2:30. It's time to get started. Your plant and have your plant that it's lacking in potassium this hour, but next hour it's going to be boron. And the hour after that it's going to be chromium. And the hour after that, we're not good at understanding that kind of specific limitation. Let the plant be in charge of its own life. Let the plant put out the exudates. The plant puts out the things that grows the organisms that start to take up the nutrients. Protozoa, nematodes, microarthropods, earthworms eat those bacteria and fungi and release the nutrients that the plant needs to take up. That's how Mother Nature has done it for the last billion years since there have been plants on this planet. All of this started with the bacteria, fungi, protozoa, nematodes, microarthropods, mycorrhizal fungi already present, cycling nutrients in the soil before a plant ever appeared on this planet. Plants came expecting that this nutrient cycling system was already in existence in the soil. So your plant is going to get all but 2 nutrients from the soil. So what are the 2 nutrients your plant gets from above ground? Nitrogen? No. Nitrogen has to come through the root system. Your plant cannot take up the form of nitrogen that exists in the air. N2 gas, not something your plant can grab. It's got to go into the soil, has to get fixed, and it's taken up through the roots as nitrate or ammonium, depending, valence. So work done by John Stark and from Cedix in France, proving perennial plants require ammonium, not nitrate. Weeds require nitrate. So nitrogen comes to the soil. What are the 2 nutrients from above ground? Carbon, as CO2, and sunlight. Does your plant need oxygen? Yes. Not as a nutrient. Conditions in the, around the plant has to stay aerobic, but the plant doesn't really need oxygen. Because remember what your plant is doing? So your plant above ground, CO2 and sunlight. Sunlight is energy. So how is your plant going to put that sunlight into a stored form? How does it take it up and put it inside the plant? Photosynthesis, which means we're going to take one carbon dioxide plus another carbon dioxide, and we're going to turn this into organic matter inside the plant. Organic matter. When we use that term organic, what do we really mean when we mean organic? When we say organic?
Carbon-based.
Carbon-based. What does that really mean? Long-chain carbon. A lot of people will say anything that's got carbon in it is organic. CO2 has carbon in it, doesn't it? Not organic. This is an inorganic form of carbon. This is not organic. It contains carbon. So containing carbon is not an adequate definition of organic. It has to be in a carbon chain. And that's what photosynthesis does, is to build a carbon chain. So this carbon from this carbon dioxide and that carbon from the second carbon dioxide are going to get bound together. The energy to build this bond is stored sunlight. That's energy. So carbon chains, organic. And as we're dealing with a carbon chain, now it's an organic material. So here's the simplest form of sugar, a 2-carbon chain. So we're looking at acetate, which would be the typical kind of sugar that we're dealing with here. So carbon from one CO2, carbon from the other, bound together by sunlight. What's the waste product here? Water. Oxygen. So we're making 2 oxygens. And that's blowing off as a gas. Your plant requires the biology around its body to be aerobic, but the plant doesn't actually need oxygen. Yeah, okay. Does it have to have oxygen in some form within the organic material? Yeah, so there it does take off of oxygen, but that oxygen comes through the root system, not as a gas in the air. Pardon?
What about at night? Is there an oxygen exchange?
Yep. There's a dark cycle on some uptake of oxygen that can kind of occur there. Yeah. So and it depends on the kind of plant. But photosynthesis really doesn't need oxygen. And certainly root systems don't have to have oxygen as a nutrient. They have to be in an aerobic condition. So isn't it subtle? Isn't that kind of feel the difference there? Kind of important. So above ground, our plants only get 2 nutrients, sunlight and CO2. Everything else has to come through the root system. So how important is the above ground part of the plant? Who cares? Because almost everything comes through the root system. When we play the scientific game, what's the most important part of your body? Answer that one. You know, scientists, we always got to go, what's most important? Now, so if you're a scientist, what is the most important organ in your body? Your brain. Right? You can take all the rest of my body away. As long as you leave me with my brain, that's the most important part, right? How long am I going to stay alive if you just leave me with my brain? Did you see where we do this in science all the time? What's the most important thing? Yeah, the most important vitamin is, right, just try to live without all of them. Right? So let's go back in time. When you were a child, when I was a child, what were the 3 most important nutrients for plants? Like, right, just try to grow a plant with just NPK. Not happening. Because we have to have carbon. We have to have sunlight. We have to have, well, by the time I was started into high school, how many essential nutrients were there? There were about 12. So back when I was a child, 3 important nutrients. By the time I was in high school, 12. By the time I got finished with college, it was up to about 24. When I finished graduate school, it was 36. Today, plant physiologists, how many essential nutrients are there for plant production? We're up to 42. It's the answer to the universe. 42. Is arsenic one of the important elements in growing plants? In that list of 42 elements today, arsenic is not included. What happens if your plant has no arsenic? It's just this little pile of green, slimy stuff on the surface of the soil. How about you? No arsenic? Is arsenic an essential nutrient in your body? Absolutely. No arsenic in your body, your nerves don't function. You're just this little quivering jello on the ground. Is arsenic an essential nutrient? Just try to live without it. Now, can you have too much arsenic? Yeah. Can you have too much oxygen? Yeah. Can you have too much water? Can you have too little water? So everything, every nutrient, every chemical in the periodic table, we probably require some amount of it. Everything on there, probably essential nutrient. We've got to have a little bit. So what we really have to think about in all of this is the right amount. So think of this as the Goldilocks principle. We have to have enough, but we have to have enough of everything. If you only have your brain, you're going to be dead. What if you're a romanticist? What's the most important organ in your body if you're romantic?
Heart.
So great, I can take everything away but your heart, and you're not going to be alive. You need everything functioning together. It's holistic. We're talking about everything has to be there in the right amount. Every nutrient, arsenic, copper, boron, everything, lead. You even require some lead in your body or you won't stay alive. But not too much. Not too little, just right. Goldilocks went into the 3 bears' house, sat down at the table, picked up the first bowl, and, whoo, too hot. Ugh. Next bowl, too cold, yuck. Next bowl, just right. Perfect. She's all up. She goes into the bedroom, lays down on the first bed. Ooh, too hard. Next bed, too soft. Next bed, just right. So she gets a good nap until the bears come home. So just right. Everything has to be in the proper balance. So how does your plant make certain that it's getting just the right balance? Who has to be in control of that? You? You have no clue what your plant requires right now at this second. There is a limiting element. There is something preventing that plant from growing any faster. But we have no clue what it is. So let's leave it up to the plant to figure out what it requires. And it does that by putting out the exudates, growing the bacteria and fungi, solubilizing the nutrients, protozoa, nematodes, microarthropods, eat them, make the nutrients available in your plant stage. Thank you. No limiting nutrients. So when we're talking about does something taste good, it's the balance of all those nutrients. It's the balance of the vitamins, of the organic materials, with all the proper nutrients and the proper balances that makes that plant material taste good. If we're overloading that plant with too much nitrate, too much phosphate, too much sulfate, too much, or not enough, the plant is not going to be in balanced growth, it's not going to be healthy, it's not going to taste good. So start trusting your tongue. If it tastes bad, it is bad. Spit it out, don't eat it. So we need to do some more testing in the world of comparing conventionally grown foods with organically grown foods, or sustainably grown foods, or biologically grown. What do we call it when we get the biology in there right and the plant's getting all the nutrients it needs in the proper balance? Do we call that organic? Do we call it biological? Do we call it nature farming? Do we call it natural agriculture? Do we call lots of different terms? organic, biodynamic, or do some of these systems get that wrong? How many times have you bought something that's grown organic, it's got the organic label on it, and you take a bite and you go, blech, doesn't taste good. Well, they're organic by substitution. They're not organic at the heart. So can we screw up even in the organic world if we're not looking at the biology and making sure that we've got the balances right? So really trying to understand this, if we get the biology right in the soil, here are the benefits. We're going to have a huge increase in the amount of water we store in the soil. As long as we can get our root systems going deep down into the soil, you really do not have to worry about water. Unless you're in a part of the world that hasn't gotten water for the last, 10, 15 years. I think we're safe here in Hawaii. So build structure, hold the water that you get. We go over to places like Saudi Arabia, real fun place to visit, especially as a woman. Go to desert systems in Africa, and we've gone on to stone desert. Go out there and get the proper biology. Yeah, you got to do a little tillage. You've got to build water holding capacity. You got to get the biology in the soil, plant some plants. So you can go from stone desert to oasis within a year, where we do not have to put water on to grow orchards of date palms or palm trees or you name it kind of plant system that you want, from stone desert to functioning oasis system where the system is bringing in its own water. As long as you got dew fall, we can convert deserts into productive systems. Been there, done that work with a lot of the permaculture guys. So get the water holding capacity back within your soil, reduce water use. In the desert parts of Australia, we have reduced water use in table grapes by 70% in the first year. In other systems where they haven't gotten the biology quite right in the first year, we reduce water use by 30 to 50% in the first year. Just start getting the proper biology back into the soil. But the critical part is organisms into the soil. If you're not doing anything to assess the biology, if you're not looking to make sure that you're getting organisms back in there, a lot of times people just aren't successful. So they say they're putting compost out. Did they measure the biology in the compost? No. Then they're going to fail a great deal of the time. They're using compost tea. What's compost tea? You're taking a compost that has, hopefully, all the right biology in it, bacteria, fungi, protozoa, nematodes, microarthropods, mycorrhizal fungi maybe as well. We're going to take that compost and we're going to use water to extract the organisms from that compost. Bacteria make a lot of glue, and they glue themselves to the surface of their organic matter. There's actually a glue layer around them. So when you're thinking about bacteria on the surface of sand, on the surface of clay, on the surface of your silt, or any organic matter, if that's our surface, so plant material, or sand or silt, that's our surface. A bacterium, as it starts to grow, makes a glue layer that binds it to that surface. So glue, there's the bacterium itself, and the bacterium has to hold itself on that surface. What if it's not glued to the surface? What if it's not bound? Well, the next time you get water moving through your soil, where's your bacterium going to go? It's going to wash off. So just think of that little bacteria going, no, don't take me away from my candy, no! Poor little guy's going to die. So it's not going to allow that to happen. So it glues itself there. When we're talking about aerobic bacteria, what's the pH of the glue? It's always alkaline. So when we're dealing with glue, huge numbers of bacteria in a typical bacterial-dominated soil, we're looking at 10 to the 12th power number of bacteria per teaspoon. Per teaspoon, or a gram of that soil. Each one of which is making a glue layer around its body to bind and glue itself to the organic matter, to the soil particles in that soil. Think of the alkaline situation. What is that effect going to what effect is this going to have on the pH of your soil? Bacterial dominated, where there's more bacteria than anything else, that soil pH is going to be greater than 7. It's going to be alkaline, above 7. If you're in a bacterial-dominated soil, the pH is going to be alkaline. Well, that's right for growing what kind of plants? It's great for growing your vegetables. It's great for growing grass. It's great for growing weeds. So, you might want to be paying attention to that. What if you want to grow really good vegetables? What's the balance of bacteria and fungi? The bacteria alone, your soil's pH is going to be 7. Remember? Aerobic. What if we go anaerobic? As anaerobic bacteria grow, they don't make those glue layers. You cannot build structure in your soil when you're dealing with anaerobic bacteria. Anaerobic bacteria don't make the glue. So now what pH is going to occur because you've got anaerobic bacteria growing? No glue. No alkaline materials being made. What are they making? These guys make a whole bunch of really nasty organic acids. So the kinds of acids that they're making are things like acetic acid. What is acetic acid? Vinegar. What's the pH on this? At high concentrations, when you're dealing with pure acetic acid, what's the pH? 2. The pure stuff. The longer you're anaerobic, the more acetic acid these guys make. What's going to happen to the pH of your soil? Butyric acid, only made under anaerobic conditions. What's this stuff? We don't have many organic chemists in the room. No, shouldn't expect that, right? Butyric acid, sour milk. Only under anaerobic conditions, when your milk's left on the counter, goes anaerobic. Bacteria start growing so fast in the milk, using the protein, the lactose, the sugar in there. They start growing so fast, they're using up oxygen faster than oxygen can diffuse into your milk. Now you've got the sour milk taste. pH 2. Why do you not drink sour milk? Well, it tastes horrible. The pH is pretty far down there. And if we go anaerobic and there's any kind of pathogens in that milk, what are you drinking? You could be killing yourself. Because only under anaerobic conditions is E. coli, Salmonella, Shigella, Pasteurella, all those human pathogens start to grow. So have human beings learned over time not to drink something that tastes bad? Yeah, because we've learned the probability of huge problems is significant if you're not careful.
Are all pathogens anaerobic?
Almost all of them.
Almost all.
There are some skin diseases that are aerobic, but not very many, and they're usually pretty easy to deal with.
So go see aerobic.
Aerobic, yeah. The more you can keep stuff aerobic, the more likely you are to select against those diseases. So butyric acid, drop in the pH. Another one of my favorites, valeric acid. Wonderful. Doesn't it sound kind of nice? Valeric acid. What's that smell? Vomit. Only produced under anaerobic conditions. Again, pH of about 2 of the pure compound. The pH is about 2. So bad smell, low pH. Putrescine, another one of my favorite anaerobic products. What do you suppose that smells like? The smell of decaying flesh. Ooh. So you walk out to a composting operation, and you smell putrescine. What is that telling you?
Anaerobic.
Anaerobic. It ain't compost, is it? You walk out to a composting operation and you smell a mixture of putrescine, ammonia, vinegar, sour milk, and vomit. Now, is that really yummy? What that mix? What we used to call in the soils world sour soil. When Steiner started talking about sour soil or sour, that's what he's talking about. It's all these anaerobic compounds that are made. When we go anaerobic, what's happening to the pH? We're heading for the little number. The only way to get a soil or a material that has a pH less than 5 is for you to go anaerobic. How many of you have soils that have a pH down around 4, 4.5, 5, 5.5? The only way they have had that happen is you're anaerobic. So, start learning what the stink smells mean. Mother Nature's sending you a message. Do you have to take your soil into the laboratory and have them measure pH in order for you to know this is anaerobic? Dig down in there, pick it up, and smell it. Or taste it. Yummy. So, now you start to understand what these odors mean. It means you went anaerobic. It's not compost. Don't buy that stuff to put on your soil because what are you doing to your soil? So anaerobic, the only way to get really acidic is to go anaerobic. And you're making lots of things that are going to be very detrimental to your plant. Now, we talk about bacteria. How about fungi? If we're talking about aerobic fungi, these fungi help hold aggregates together. But what they're actually doing is taking their strands. Fungi grow as long threads, long strands. And some of these can be, quarter of a mile, half a mile. They can be very long. People have followed fungi through the soil for long distances. The largest organism on this planet is fungus. The current winner of the largest organism on this planet lives in Washington State, on the mainland. So Paul Stamets spent a couple years documenting that this fungal species, single individual, is about 20 miles across in every direction. It lives in a forest, oak grove forest, in Washington State. The fungus itself is down maybe just a couple inches into the soil to as far down as 15 to 25 feet down into the soil. This single individual, as documented by PCR by doing the DNA analysis, this single individual is on the order of about 5 blue whales, largest organism on the planet. So, huge, long strands, lots of branching, grows out, gets eaten by its predators in the summertime. Once it's done getting eaten, then fall happens and it expands out again. So lots of growth, lots of nutrient cycling going on here. These strands bind themselves around the microaggregates built by the bacteria. So the bacteria are gluing, there's our bacteria in there, gluing the materials inside the sand, the silt, the organic matter glued together by the bacteria. The fungus comes along and wraps all of this, pulls it together into the macro aggregates that you can see with your eyeball. But fungi don't make a lot of glue. They make a little, not a lot. Their major contribution to soil pH is the fact that they make organic acids, but they are not anaerobic. They are aerobic organic acids. And these organic acids will never, no one has documented an organic acid made by fungi that has a pH less than 5.5. So the organic acids made by fungi between 5.5 and 7. So now, if you have a fungal-dominated soil, where we have more fungi biomass than bacterial biomass, the pH of that soil is going to be between 5.5 and 7. Who controls pH in your soil? The organisms. Who's getting the fungi to grow or the bacteria to grow? Rough land. Who's in control of pH in your soil? Why would we ever have suspected that it would be otherwise? Who's trying to sell you what? And so how do you control the pH in your own soil? When it gets compacted and it goes anaerobic, what's going to happen to the pH? Really low. So get oxygen back in there. Now, is it going to be fungi growing more or bacteria growing more? Or what plant are you trying to get to grow? So we've got a little bit more we got to understand here. It's not just microorganisms. It's what's this balance? As we get the microorganisms growing in there, what's the proper balance? So we've got that little bit more information to go.
Just blurt it out. You said no need to rotate crops.
There is no need to rotate crops. Yeah, because why do you rotate crops? What's the point and what's the purpose of rotation?
You exhausted the nutrients, so you're going to get to a different one.
Yeah, do we really exhaust nutrients in our soil when we stay in the same crop time after time after time? Yeah, probably what we're exhausting is diversity of the microorganisms. So by growing one crop all the time, we're only putting in limited food sources. So how can you and I fix that particular problem? Well, putting compost.
Or tea.
Because then we're putting, or tea. So I was going down that, I was going to answer that question, what is tea? See how I get distracted? Did you notice that already? So you might have to drag me back and get me to answer something I was starting off on. Compost. We're going to take that compost, a small amount of it, and put that into 100 gallons of our water. So we're going to take 3 pounds, maybe 5 pounds of our compost, put it in the water, let's bubble some air up through there. So we're going to keep this water all nice and aerobic. We're going to use the movement of the water to rip the organisms out of the compost. Those bacteria glued. We're going to have to apply a lot of pressure to take those bacteria and rip them off that surface. Have to have enough water pressure to rip. Think of all those little bacteria in there going, no, not if I know. Because we want to get them into the water. We're going to get the fungi pulled out, the protozoa, the nematodes. We want to get all those microorganisms pulled into the water. Any of the soluble material in our compost is going to be pulled out into the water. Why is compost brown?
Really because of the humic acid.
Yeah. Remember that all the starting materials that went into our compost, it wasn't brown. Where'd the brown come from? So, manure. Is it a nice dark brownish color? Green plant material starts out green. Dry brown plant material, wood chip. It doesn't usually start out a nice rich dark brown. It's only as the bacteria and fungi decompose all of that plant material, and they take those simple carbon compounds and turn them into ever more complex carbon compounds. Fulvic acid, humic acid. Fulvic acid, what color?
Yellow.
Yellow. Tan, honey color. Humic acid, dark brown, rich color. So, can I point you to a place where you can always go to get a color chart? How do you know that you've got good humus? Well, your color chart exists in the grocery store. Go to the grocery store to the chocolate bar counter and buy a chocolate bar that is at least 70% cocoa. And that is the color of humic acid. If you've got good compost, that is the color of that compost. You walk out into a compost pile and it's black. How many of you know what color black is? So your sweater right there. If that's the color of your compost, it ain't compost. It's putrid, stinky, smelly, black, horrible stuff. Kill your plants. So not the color of compost. You know, so you go back and people are always talking about black soil is so good. Wrong. It's anaerobic. You got some really nasty stuff in there. So what is the color that we want? 70% cocoa. Go out looking for compost, bring your chocolate bar with you. So here you are, you're out in the compost pile, and on the compost, the municipal composting operation, and you pull your chocolate bar and go, ew, that's black. Not on that. That's kind of a light brown, tannish color. Doesn't have biology I want. Go to the next one. Go through the whole you go to every composting operation on the island, and what you discover is none of them make compost. They all stink to high heaven. They are the wrong color. So see, when you get done at the end of that big long trip and you realize no one's making decent compost in our municipal composting operation, at least you got some comfort food. Take a bite of your chocolate and you feel better, right? You're going to have to make it stop it. Or you go visit somebody like John, or you visit somebody like Leon, people who are making good compost, that have the biology in that material. So take a look at it, see that you've got the really good fungi, protozoa, nematodes, microarthropods, you've got the critters in there, a handful of that into your compost pile when you start it. And now you've got the inoculum of local, indigenous, Hawaiian microorganisms. How do we know that they're good indigenous Hawaiian microorganisms growing in your compost? So, you know, and it's, think about all the whole diversity that we have to have. You know, it's not just one or two species of fungi. It's not one or two species of bacteria. Every bacterial species is selected by temperature and by moisture and by calcium concentration and potassium and sodium and magnesium and you name it, all of those microorganisms. So when you realize every time you shift temperature by about 5 degrees, all of the organisms that were awake and functioning at 70 degrees, all of those guys are going to go to sleep. Not functioning, not doing their job by the time you get to 75. By the time you get to 80, all of these guys are asleep. It's a whole different set of species that have woken up and started to do their process. How about 85? How about 90, 95? Do your soils ever make it to 100 degrees? Yeah, some of them, especially if they're bare. Now, how much sunlight is hitting that soil surface? How hot are they getting? Do your soils ever get down to 65? But depends on where you are. Now, Pennsylvania, do our soils get to 65 degrees? Oh, yeah. Yeah, the last time we saw 65 degrees about 3 months ago. So right now, Rosedale, our soils are sitting down there at about 40 degrees, 35 degrees. Are there active living growing organisms at 35 degrees? Yeah. Oh, yeah, no problem. How about at minus 10 degrees? Oh, yeah, no problem. They live in frozen soil. How about molten lava? Are there living, active, growing organisms in molten lava? Oh, yeah. Just because we never bothered looking doesn't mean they're not there. So the people at the Macaulay Institute in Aberdeen, Scotland, they have extreme condition laboratories, and they've been looking at the growth of microorganisms in molten lava. They are there. They are functioning. So, species. How many different species of bacteria do we need to have in your soil? Well, it depends on the temperature. We've got to have several thousand bacteria here and another several thousand different species here and several thousand other species here. But now let's impose moisture. There are some microorganisms that can grow at 2% moisture, but they're going to be a completely different set of species when we're at 10% moisture. And all of them go to sleep when somebody else starts functioning when we hit 20%, or 30, or 40, or 50. How many species? We start looking at soils using PCR, DNA method. And in a good, healthy soil, what we typically find, we're coming out of Duke University, coming out of Cornell, coming out of Auburn, for example, a number of different universities saying that there ought to be, on average, in healthy soil, 75,000 species of bacteria per teaspoon. Compost, at least 75,000 different species, doing DNA analysis. When we start looking at fungi, we probably need to have somewhere around 25,000 different species of fungi per teaspoon. In a tropical forest, in 1 acre of tropical forest, how many species of bacteria? James Tichy at the Center for Microbial Ecology at Michigan State University suggests there's over 1 million, 1 acre of tropical forest. Work being done by Peter Vitusic here in Hawaii, suggesting at least that many in an acre of your tropical forest. Okay, how many acres of tropical forest on Hawaii? How many species of bacteria on this island? A couple million? We haven't even begun to understand this. How are you going to use this knowledge in your own work? So what we have to do is just maximize diversity. We want as many different species of food going into that compost pile. We want lots of different kinds of green stuff. We want lots of different kinds of high nitrogen. We want lots of different kinds of browns. So we're maximizing the food for all those different species. We want the temperatures in our piles to start out at ambient and go up. Do the process of getting rid of the pathogens by getting to high temperature for long enough. How do you make sure you've killed all the pathogenic organisms in your compost? Well, you either let it go through a worm, that kills all the pathogens, or you got to get the temperature above greater than 131 degrees Fahrenheit for 10 to 15 days. You're going to have to turn it a few times in there so it doesn't go anaerobic. How do you know it's going anaerobic? Well, when the temperature starts getting around 160, 165 Fahrenheit, you want to turn it because the organisms are growing so fast, they're using up oxygen. How does your compost generate temperature like that? What's causing temperature to increase that rapidly from, 80, 85 degrees up to 160? Okay, you're killing pathogens, for sure, but what's causing that increase in temperature? Yeah, it's the growth of the bacteria and fungi. As they reproduce really fast, well, think about yourself. When you perform reproductive acts, woohoo, does it get a little warm? Same thing for critters. So as they are reproducing, as they're going from 1 bacterium to 2 bacteria in 20 minutes, another 20 minutes, those 2 bacteria become 4. 20 minutes later, those 4 become 16. 20 minutes later, the 16 become 84. 20 minutes later, we're going from 1 bacterium to 1 million bacteria in 24 hours. Heat is being generated, and that's what's causing the increase in temperature. So you can get rid of pathogens either way. Let the earthworms eat it, because passage through an earthworm's digestive system kills off the pathogens, or thermal compost, you let the temperature kill them. But you got to get high enough, long enough. Well, what if you're up at, 145 degrees? Well, then you'll only have to be up at high temperature for about 10 days. And you will kill the pathogens off. What if you hit 155? What if you hit 165? Your pathogens are going to be taken care of in about 24 hours. We may not have actually done this kind of work with compost yet, but in the food industry, these are well-documented numbers. Maintain pumping at high enough temperature for 24 hours, and you've taken out your pathogens. E. coli, Salmonella, Shigella, Pasteurella, they're gone. If you're only getting to 131, 15 days. Everything has to stay above 131 for the full 15 days. What if you can get it a little higher? You don't have to keep it up that long. Regulation. Okay, the regs have not kept up with scientific data. We got a little problem there. We got to be working on this, hopefully at Rodale. We're going to put all that data together for the composting industry so they understand what we've already discovered, about 30 years ago in the world of food science. But we got to have the data. So temperatures, moisture, calcium concentration, nitrogen concentration, all of the factors that go into determining how many species are present in your compost, in your soil. And we just have to maximize diversity. You and I do not need to know that we've got Mary, George, Bob, all of the different species, bacteria or fungi present. Just get as many as you possibly can. Constantly, always, be maximizing diversity. So, see, for the rest of your life, none of you will ever go on vacation again. You're going to go on bacteria and fungi, protozoa, nematode, microarthropod collecting tours. So every place you go collect a little good biology everywhere on the island of Hawaii, or maybe even going over to Maui or any of the other islands. You always bring your little plastic bag. When you start looking in the soil, ooh, look at this really good fungus. Take a little tiny, just a little bit, a teaspoon into the plastic bag, bring it home, dig into your compost pile, deposit your inoculum. Native microorganisms, but maximize the diversity. So when we're talking about natural farming, they've started this process. You get your rice, and you get inoculum of the organisms, and you get all those real good organisms growing on that rice. Woo, nice diversity. But is it everybody? Is it everyone you need? It's a good start, but it's not everybody. So, can you do step 1, step 2, step 3? Yeah, but getting the life back into your soil is a 12-step program. You didn't know you are now becoming part of the organization called Pesticides Anonymous. More pesticides in our life, but it's a process of building that biology. And really, you're going to do it in your compost pile. So you learn to make really good compost. You may do a couple of oopses. Start trying to compost, get that recipe of really good organisms going in your compost pile. So you start building diversity. It may take you a couple of years to get the full diversity back into your soil, so you will build the structure that you require, and you build that structure down as far as it possibly can go. How deep can roots of banana plants go? How far down can you find roots of a banana going down? Now, if you're a chemical person, you would probably say, banana roots don't go down more than just a couple inches. That's because they're really sick, unhealthy, and going to die pretty soon. How far down can banana roots go? We've got them to 25 feet. So the most productive banana plantation in the country of Australia is one that we've been working with in Coffs Harbour for about the last 12 years. He has won the best banana production facility, that award. They have a contest in Australia, and he's won that award every year for the last 4 years. And he uses no pesticides, no inorganic fertilizers, nothing toxic going on to his land. All he does is make compost. And that's all he does. Compost and compost tea. Extracting the organisms into the water and applying those organisms onto the foliar surfaces to maintain health above ground. Cover all of those surfaces with the proper biology. And he actually only applied compost tea or compost extract for the first 2 years, and then he no longer had to do it. When I first started working with Graham, Coffs Harbour in Australia, I went out on his property, walked out into his banana plantation. I kind of looked around and went, where are the bananas? Those look like papaya to me, because his bananas have stem coming up and two little leaves coming off like this, flat. I was like, that is the thickest papaya I've ever seen in my life. No, that's bananas. You're kidding. That is just not true. He had beetles that were attacking the root system and just eating all of his roots off and chewing up into the stem of the banana. He had not made any cakeys for like 2-3 years. And standing at the edge of his property looking up the hill, I could see his house. There, you know, that was a good quarter of a mile away. Banana plantation? I don't think so. So we got out there, we cleared the grass away from his plant. So out to the drip line on that plant, we cleared all the grass out, applied compost at about a good 2 to 3 inches all around the poor little sick, dying plant. And in about 2 weeks, all the beetles were gone. The roots started going out into that compost, and the plants started growing. Within a month, we had 5-6 leaves on the plants, and plants just took off. A year later, he had normal production of bananas on his plants. All of a sudden, standing at the edge of his property, you could not see the house at all. It was 6 months for recovery to occur if he got the proper biology out there. So it's taken the compost, applying the organisms. He got rid of the fungal diseases on the surfaces, so no more of the black fungus growing on the leaf surfaces. Got rid of that completely. Phytophthora, thing of the past. You know, so a real success story. Today, he's the most productive banana production system in all of Australia. So how rapidly can this occur if you get the biology back? There's the trick. How exactly are we going to get those organisms back into the soil and help them start building that structure? His banana roots go down 25 feet. We've had some of the worst droughts ever in Australia. For the last 35 years, Australia has been suffering drought. The last 15 have been desperate. Coffs Harbour actually shut down all of the government offices in Coffs Harbour because we ran out of drinking water. There was no drinking water to be had. But he had no problems growing his bananas. He doesn't have to apply water. No irrigation is required, even in the middle of a 15-year drought, because the root systems go down deep enough to get the water he requires. Question?
Yeah, you said you removed the grass. That was primarily just to kind of agitate the soil and...
No, because what's the balance of fungi and bacteria on grass as compared to bananas?
But bananas are grass? Or
Bananas
it's
still
kind of
a really big grass plant.
So you're saying it needs the fungi much more than the other grass, so you were actually trying to radically modify the environment around it. Okay.
Yep. So bananas actually need to be on the fungal side. Not massively fungal, but they need to be about 2 to 5 times more fungal than bacterial. Well, you still need a million bacteria per gram of soil. It's just that now you're going to have 3,000 to 5,000 micrograms of fungal biomass in that soil. That means when you dig into the soil around any of your trees, you ought to actually be able to see the fungal strands within that soil. Or the system is not to the place where you're going to be able to grow healthy bananas and have the root systems down as deep as they need to go. So, just a question?
So you were talking about water retention. Would you be able to use the compost system like that on young plants, seedlings, and not have to irrigate?
Mm-hmm. So when you plant a plant, so even if the seedling's only got a root system like that, we want to make sure the soil around it, the biology is building that structure. So that root system, as soon as it goes into the ground, zoom, down it goes. Because we don't want, you usually have grasses or something up here that are competing with your plant for nutrients. So we want to get that root system growing down as fast as possible. Way to build structure is to make sure we have that biology. So putting compost down there, get that biology, build structure. It can hopefully go faster than the root system of your plant is going to move down, requiring the well-structured soil so it can just grow as deep as it possibly can go as fast as possible. How do you tell in your soil whether you've got compaction problems? Metal rod. Go and grab a nice long metal rod, get a pressure gauge that you're going to just hold on the top of that metal rod until the point of your metal rod sits on the surface of your soil. The pressure gauge right on top of that rod, and start pushing on the pressure gauge so you can read on the pressure gauge how much pressure do you have to apply to get that metal rod to move through the soil. And if it takes more than 100 pounds per square inch pressure for you to move that metal rod through the soil, your roots are not going to grow through that soil. There's been a lot of work done at Cornell University, so they have a number of different plots of that graphical information. What is the PSI, pounds per square inch, where root systems can grow normally into the soil? It has to be less than 100 PSI. By the time you get to 150 PSI, most of our vegetables, grasses, can put their root systems through soil that's more than that compacted. By the time you get to 300 PSI, there's almost no plant alive that can push its root systems through that soil. You are going anaerobic, you're going to be making disease-causing organisms that will attack and consume your plant. So there's the easy way. Push that. You can get penetrometers. So penetrometers. They're about $300–400, where they come with a pair of handlebars that holds the pressure gauge, and you just push on the handlebars, push that rod down through the soil, find out where your compaction is. Find out if you've got a problem. There are a lot of folks here in Hawaii. When I was working with Chuck Warner over on Maui, one of the original organic farms in Hawaii, he was always, oh no, I don't have any compaction at all. Yeah? Prove it to me. Went out there with the penetrometer, and about that far down into the soil, we were at 300 PSI. So, good compost. Let's make good compost. Now, that was a challenge, because Maui, how much rainfall do we get over on the Hana side of Maui? Oh, that was a little scary. The most compacting problem that he's got, the thing that compacts his soil most of anything else, is rainfall. Nice, big, have you noticed how big the drops are in this part of the world? And they're coming down and one of those hits you in the head? Oh, that hurts. So imagine what it does when it hits your soil surface. That is compacting the soil. Where does it compact it? At about 3 to 4 inches down into the ground. Now here's the surface. Now until it gets really, really bad, are you compacting here at the surface? So where you're going to start compaction, typically just with rainfall, is about 3 to 4 inches down. So how far down in the roots of your plants going? Go home today. Dig up your grass and your lawn. How far down are the roots going? How far down can roots of zoysia go? The typical grass that you've got in your lawn, how far down can the root systems of zoysia go? 10 feet. And if they're not going 10 feet, Mother Nature's sending you a message. You got compaction. How do you fix that problem? Biology. Back into the soil. So we want to see these nice fungal strands. You can see where some of this is a little out of focus. Looking through a microscope, 400 total magnification. You can see those nice fungal strands. Some of it's a little out of focus, so you're going to remember to focus with your fine focus. So you're actually following along. Fungal hyphae are always uniform diameter all the way along the length of that strand. Okay, they can branch. Okay, the branch does not have to be the same diameter. Typically it's the same diameter, but not always. Really good beneficial fungi are going to be dark brown in color. They're going to be colored, maybe tan colored. If they're wide enough diameter, they may still be okay if they're colorless. The wider the diameter, the more likely it is a beneficial fungus. Color of any kind of beneficial fungus. The bad guy fungi, the disease-causing fungi, narrow diameter, 2 micrometers clear, bad guys. almost invariably bad guys. So how many of you can find the real narrow diameter fungus, clear, colorless in this picture? It's a little out of focus. At center stage, can you find them? Right there? A little out of focus, but very narrow diameter. Look at that diameter as compared to that. Or even this guy over here, or even this colorless, but look at the diameter. There's the disease. The rest of them are beneficial fungi. So we got a disease-causing fungus in this compost or in this soil. Does that mean this is bad? Growth of that disease-causing fungus never happens, because the beneficial organisms will wipe out that disease-causing fungus. What if we treated this with a fungicide? Who's going to survive the fungicide? Disease. Yeah, the fungicide wipes everything else out. The beneficials are gone. The only thing that survives is the Pythium, because there is no fungicide for Pythium that's effective when we're dealing with soil. So, what are you actually going to end up with if you happen to use a fungicide to try and kill the fungal disease? You're going to end up with fungal disease. Amazing how that works. Look at all the nice aggregates in here. So here is our bacteria. See all those little grainy guys in there? Those are our bacteria. Gluing all of the sand silts in there, the organic matter. What color is this? Nice background rich. Good humic. See the tan color in there? Fulvic. We've got some omics. We've got some carbs. We've got some sugars in there. It's a clear colorless stuff. So the glue that the bacteria are making to hold everything together. Then the fungal strands would have been binding all of these micro aggregates together in a macro aggregate. Of course, we shook this sample and kind of broke some of that up so we could see the fungi, so we could see the bacteria. How many bacteria in this picture? How would you like to count them? There's millions. There's an individual bacterium. There's another one. There's another. How many species of bacteria do we have right here? If it looks different, it is a different species. So we're looking at several different species of bacteria right in here. Now you start looking, I don't even want to think about counting all the bacteria in that microaggregate. I'm going to dilute it some more. So I'm going to take 1 milliliter of that 1 to 9 dilution. I'm going to take 1 ml of that out, dilute it another 1 to 10. Maybe dilute it another 1 to 10 until I get all those bacteria spread out so it's easy to count them. We're just going to dilute so we can easily count those bacteria and figure out how many bacteria we have. Figure out what's that fungal. How many fungal strands do we have? So we make it easy to do these estimates. So bacteria and fungi in the soil, how do you tell if they're good or if they're bad? Fungi, it's easy. Look at diameter. Look at color. Good guys, dark. Color. Wide diameter. Easy to tell. I'm going to show you some results of using compost teas because people often don't believe me when I say as we adjust the balance of fungi to bacteria, as we go from completely bacterial-dominated weed production systems, as we start to get the fungi back into the system, we get rid of all the weeds. They will not occur. The seeds will not germinate. They will not grow because they won't have the proper form of nitrogen or phosphorus or sulfur or magnesium or calcium, blah, 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 blah. And so we get rid of the weeds just by managing biology. So Ian Smith was in Tasmania. He's been an onion grower for years and heard about all of this biology, how do you get the biology out, so he started making compost tea. Into one of his 150-acre paddock fields, he applied 3 applications of liquid compost, or compost tea, where we extracted the organisms, a little bit of food to grow fungi, help the fungi along, and real fungi out so they were adequate. So 2 applications of tea before seeding, and then 1 application after the onions were put into the system, as compared to the regular conventional system where he's applying Roundup. His reduced herbicide rate, he's talking about that because in the compost tea paddock, he has been using herbicide up until the day he applied the compost tea. So it's for the last month and a half since he applied compost tea, he's been using herbicides up to that time. So, let's look at some pictures that he sent. There's the conventional system. Does he have a little weed problem here? That's with 3 applications of Roundup. And look at the weeds. Can you find the onions? It's almost kind of hard, isn't it? Oh, yeah, you can see the onions up. The onion growth is being affected by the weeds. Because do we have Roundup ready weeds in Tasmania? Yes. The Roundup isn't doing much here. So how do you deal with that kind of weed situation? We're going to show you the root system on the onions. So we dug up onions from a number of places, or Ian dug up onions. I'll show you some of those root systems in the conventional system. Now, let's look at the biological system. See any weeds? There are no weeds in the picture. 3 applications of compost tea, and we have dealt with the weed problem. Look at the onions. They're bigger. I'll show you the root systems in just a few minutes. Now, that's 150 acres. And just to prove to you that it's not just this one picture, let's look at the field in all kinds of directions, and you can see that weeds are not a problem in this field. Okay, it's not perfect, but this is certainly not the problem and not having an impact on the growth of the onions, where all we've done is applied 3 applications of compost tea. Compost tea, how much does it cost to apply it? It's 3 cents an acre. 3 cents. Versus the Roundup. What did 3 applications of Roundup cost him on this property? So we're talking about thousands of dollars per Roundup application versus a couple $100 applying the compost. There's the onion root system. In the conventional system, you can see that the root's going down about that far, and we've got disease on it. You can see some of the brown, necrosis that's happening on that root system, and you can enjoy the weeds coming up in that system. Now, let's look at the roots. So that root system is down at about 2.5, 3 feet. Same age, those onions were planted on the same date, and look at the difference. Significant improvement in yield between these 2 different systems. Now, in this paddock that he applied the compost tea, what he did here was not put any compost tea on this one patch in the field. So did the compost tea, did altering the biology in the soil get rid of the weeds? Beyond a shadow of a doubt, there's our control. How easy is it to exit weeds from your life? So let's get cracking on this. It's just shifting from a fully bacterial-dominated soil to something where we have about an equal biomass of fungi and bacteria. Say goodbye to weeds. So if you can get it right, that's what we need to be doing. Got to make your compost first. So get the biology right in your compost, extract those organisms and put them out in a liquid fashion. Or what if you don't want to have to apply it in a liquid fashion? Well, then put compost out there. One or the other, I don't really care. Whatever's easiest, whatever's least expensive. Now, imagine putting a ton of compost per acre out over 150 acres. That's a little fun. That's kind of an expensive process. So can you see why they turn it into a liquid form? Spray it out. They already have the irrigation equipment. They just run it through the irrigation in order to put that proper biology out into the system. Question?
So what are the strategies for making the right kind of compost? I mean, that's kind of...
Let's go into that after I get done with these examples. I was going to bounce it. Well, I want to show you the successional picture, but we got to get there first. One more example. This is in pasture, so this is grazing. So the last example was vegetables, when we're looking at large acreage commercial production. Now, most of you may not have huge, enormous commercial acreage, but I hope you can see where it's easily applied to a small garden, or half an acre, or a quarter acre, 2 acres, 5 acres. Much easier if we're doing smaller areas. You don't have to make as much compost. So but now grazing systems. These are dairy that we're working with, again, in Australia. And so a couple of the different farms, I'm going to concentrate on the Whiting's farm, but the data are the same coming from all of these systems. When we first started working with these folks, their fertilizer costs were just going through the roof. 5 years ago, they were applying 450 pounds per acre of urea. By the time we started working with them, they had to be putting out 800 pounds of urea per acre to grow grass. So they have almost doubled their fertilizer applications in 5 years of being doing dairy production, because they just can't get the grass to grow. So in their chemical system, they're putting more on and more on and more on, and they're just going broke. They're going bankrupt. The insects are getting out of control. So on lucerne, which is the alfalfa, we call it alfalfa in the U.S., lucerne, they have to use insecticides to reduce the insect pressure so they can have any kind of nitrogen fixation. They had no management plan for the manure, so huge manure piles, liquid material on the farm in various places. The fly problem was just out of control. No clover in our pastures. The grass was being pulled out by its roots when the animals were grazing, so they were developing huge bare spots. They had to resow their pastures every year in order to maintain plant production. To resow a pasture costs about $2,000 per acre. So 300-acre farm, you can imagine costs are starting to go broke. Profit margins aren't good. Most animals are sick. So, farm after farm, we've got problems. So what do you do? Well, there's where you feed the animals. So they're collecting manure off these surfaces when they're bringing the animals to feed them. When they're in the dairy parlor, you collect the manure from there. What they had been doing with all that manure is just washing it out of the system into big pits that they had dug in the ground. So this is a 15-foot pit. In 1 year, they had filled up the whole pit. Can you imagine the smell? And the liquid coming off is in those lagoons, and then they use that water to put nutrients back on their paddocks, and it's killing their grass. To put that concentration of nitrates and phosphates and sulfurs back onto the pastures. So they're going downhill fast. They're going to be out of business. There's the flies. You can't really see them in this picture, but they were horrible. You know how we always talk about the Australian flute? Because you got to do that when you're around these manure piles, because otherwise the flies just are all over your face. It's really not fun. So what do you do instead? You turn your manure into compost. They've got all the dry ground material that you have to have. So from stock material, from wood chips, from paper, from cardboard. When they're getting the fertilizers, they're often coming in brown paper bags. Chip it up. Goes into the compost. So these piles, in general, are about 50% brown material. So stalks from plants, sawdust, wood chips, brown paper, office paper that they're getting from their own property, from their own operations, or leaves. So when you're down leaves from plants, going into piles, and then they're going into the composting operation. Green plant material, so they're just mowing some of their own grass, or they're mowing the weeds. That goes in as the green component, so about 40% green, and then using 10% of these piles manure straight from their own manure lagoon. The tricky part here is you got to have a turner. So what Camper Down Compost did was they bought a turner that they would bring out to the farm. So when these compost piles needed to be turned, when they hit about 160, 165, they brought the turner out on a truck. They turned the compost, and then we just drive on to the next dairy farm, turn the compost there, drive on to the next farm, turn the compost pile there. You only have to make compost once. It takes about 3 months. And then you're done with that part of the system. So when they started applying this, some fields they put on 1 ton per acre of the compost. Really bad fields, they put on 3 tons. Well, they're making their own compost, so it's right there. It's not a huge transportation cost. In those farms where they didn't make a lot of compost, they turned their compost into tea, compost tea, and applied it at 5 gallons per acre. So very inexpensive. Here's a turner, just shows you, the sawdust going into the system and turning it off, monitoring the temperature. So for about 15 days, you've got to monitor the temperature. It doesn't really, it takes maybe a half hour out of your day to be monitoring your piles on a daily basis, but it only goes on for about 15 days. In this particular town, they were putting out 3 tons per acre. Excuse me, 1.5 tons per acre. Sorry. And there's their pastures. They have not had clover like this in their pastures ever. A lot of the older growers talk about the clover being like this back when they were children, 10, 15 years old. And those growers, these dairy guys, are 70 years old. So it's been 60 years since they've had clover growing in their pastures. So the clover starts coming back. Well, you're fixing your own nitrogen fixing plant, so reduce your nitrogen requirement. So these guys, in the first year, typically reduce their urea applications by 50%. By the second year, they have reduced their urea applications by another 50%. And this year, they're using no urea at all. So each year, they have reduced their cost per farm by $100,000. Each year for the last 3 years. Does that make a difference? And look at the grass production. This pasture was grazed 10 days ago. And the grass was taken down to the crown surface. And look at how much this grass has grown in the middle of the winter in 10 days. You can put your animals back on this within 10 days. So they're looking at the root systems, they're looking at the nodule, the nitrogen fixing plant material. Look at those root systems. This is within about 3 months of the first application of the compost or compost tea. Roots are going down this far. How far down can the roots of these grasses? Highly productive grass plants, not your zoysia. That's, these guys only go down about 10 feet, but these roots will eventually get down to about 25 feet deep into the soil. Going to have to worry about drought? No. We'll never have to water these paddocks again. So over the course of 3 years, they've reduced their cost by $160,000. Comparison then of the conventional system where we put on the compost or compost tea. So at the same time of year, this was re-sowed just a couple months before this. This has not been re-sowed at all. So look at the difference in grass production. So conventional versus biological. They put the animals were out here and grazed 3 months ago. They have not been able to put the animals back on that pasture in 3 months. Look at how much the grass is growing on this side of the field. You can't put animals back on this. Not even now. This was grazed 10 days ago. You can put the animals back out there to graze once again. A difference in plant production. Now I'll just go through the year. You have to remember that this is through the winter. December is midsummer in Australia. April then is late fall, going into winter time. And just looking at the quality of that pasture. Again, this has not been able to be grazed at all through the whole time period between December and April. They're grazing animals over here every 10 days. So the animals graze, 10 days, the animals can come back. So they've increased their dairy herd because they've got way more grazing pasture. Because you can get the animals right back on it very rapidly. So reducing their fertilizer costs is just, different dairies, slightly different costs, different, they don't have abortions anymore in the animals, so they're not losing their the calves. The mama cows are being successful at producing babies, so the stocking rate has increased by 15%. They don't have diseases, they don't have insects. They've got the clovers, they've got the alfalfas back in the year. They're using no pesticides, no inorganic fertilizers, and the animals are healthier. The reduction in the veterinary bills, because the vets are not coming to visit these dairies anymore, that in itself pays for the compost production or composting. Just a couple of examples. So, one more example, real quick.
Do green manures have a place in a rotation, to increase organic matter as well for intensive vegetables?
Green manures, are they necessary? No, not at all. But you have to think about the fact of when you go out and when you till in your crop at the end of the growing season, that's really not a very good idea, because you start tilling that plant material into the soil and you slice it and dice it and harming your organisms. So what do you do instead? So if you're going to till in a previous crop, then a green manure is a really good idea, because you got to get something growing in that soil. Mother Nature abhors bare soil. She will put a weed in it. Well, and what did you just do to your biology when you tilled? You pushed it very bacterial. So what's the way around that system? Let's put in permanent understory plants. So at Rodale, what we're starting to do in our agricultural systems and in our orchards and all of our perennial systems is putting in a permanent understory plant. Real short, low-growing plant species, things that don't get about maybe 3 to 6 inches max. When we're in corn, not a problem to have something that's 6 inches tall. When we're dealing with lettuce, can't have something 6 inches tall. So we're going to go with the really prostrate things. So into our fields where lettuce is going in, we're putting in these real low-growing trefoils. We're putting in some of the prostrate clovers. We're putting in a number of different plant species native to the system, and that stays as a permanent crop. So through the winter time, we've got these low-growing plants growing. They're completely covering all of the soil. Come springtime, when we're going to plant our lettuce in there, we just strip till that strip that's going to get the lettuce, and we plant our lettuce into that system. Or corn, or soybean, or wheat, or whatever we're going to grow, that gets seeded into the system, whatever method you're going to seed it in. We typically, we're going to, just broadcast that seed and lightly rake. So a harrow works really well when you're doing something broadcast. Something that's going in a row, just strip till into that. But you leave all the other plants in place. Most growers have real problems in the springtime, because as you get springtime rain, and that rain gets on your soil, and you try to get your tractor in there, you can't. You can't plant nothing until it's been dry enough for a long enough period to be able to get your tractor into that soil, right? Never have that problem again if you have an understory plant species in there. So we don't ever have a problem getting our tractors in, and we plant exactly on the day that we had planned to plant, because our tires have plants that they're going to be driving on. We strip till, so we've got tillage equipment that has 16 rows. In it goes, and we plant our seed on the day we plan to plant, because it doesn't matter anymore. Our soils don't turn into mud. We're on our crop, on our understory crop species. So the plant goes in. How wide of strips you have to till out depends on the plant. How fast is that seed going to germinate, pop up, and start shading your understory plant? So corn, it happens about 10 days. The corn comes up and it's already shading the understory plant, so corn has no problem. Soybeans takes a little bit longer. Lettuce, you got to be really careful that your lettuce comes right on up and you don't have any tall plants, so you just got to make sure your understory plant species are the ones that will not get above your crop, whatever it happens to be. Those of you who might be interested in seeing some of the work that was done in North Dakota, South Dakota, Manitoba, there is a webinar that you can go look at on Google. So Google the name Jay Fuhrer. You have the website address, but not up on my computer. Like Heil Hitler, Fuhrer? Oh. So he is a USDA Extension Service person up on Great Plains Research Station in North Dakota. And he has worked with 4 different farms. And he's got the data from those 4 farms on that webinar. So if you Google his name, you should be able to come up with this webinar. And what he's showing is on each of the farms, it doesn't matter if it's pasture, if it's a row crop production system, if it's, whatever, orchard, whatever system. So 4 very different farms, very different crops. The idea is to get a mix of short, seeded plants. You want to make sure that you put them in as seeds. Let's make sure that they are the right kind of mycorrhizal fungi for the other plants that you want to be growing into the system. Let's make sure that they are good food for bees. So bee forage, nice thick crowns, so that weeds will not be able to outcompete them. And so they're putting something like 15 different species of plants, all seeds. You just go out and broadcast these things in that have these characteristics. And what they found in the first spring is that 3 different species of the plants actually germinated and grew. And next year, it was 4 different species, not these species that came up in the first year. It was 4 different species of the 15 that they seeded out there. The next year, it was a different set of 3 species, and the year after that, it was a different set, and the year after that, they have never repeated the same mix of species that have come up. It's always been a completely different set of species or a different mix of species. Why would that be? Because the weather's never been the same. How many of you have had the same weather pattern year after year? It's never happened, has it? Every year is unique and different unto itself. So it better not be just one species. See the problem that we have when we're dealing with monocultures of a green cover crop? Is it going to be a good year for the rye this year? Or would it have been better to put oats out? Or should you maybe have hedge your bet, put all of them out. And just make sure that you start picking species that are native and have these characteristics. Make sure you got a clover in there, that you have a trefoil, that you have a nitrogen-fixing plant species in that mix, but just make sure they're short. So what are those right species for here in Hawaii? I bet you could tell me better than I could tell you. You probably know those species better. So get together as a group. Let's go grab the plant ecologists at this university and say, let's develop the mix. So at Rodale, we are developing that mix of species. We've put them out. They've been growing. At the end of this growing season, we're going to harvest those seeds. Now we're going to sell it to people as the understory plant species for them to start doing this. Here's a business for all of you to start doing this. Start growing what's successful on your own land and turn it into a business for yourself. Get all your friends to buy it, and once they've bought it, expand it so we go statewide on this one so we can start being more sustainable. If you get these root systems of the understory plants going down, how deep? You're building structure, you're maintaining that structure, maintaining that food web, you're not even gonna have to put compost down next year. It could end up being a one-time application of compost and or compost tea if you get the understory plants going in your soil. How would you like to basically put your plants in, walk away, let them grow, and come back and harvest? Isn't that how we should be growing plants? Why do we have to make it so hard on ourselves? Coffee. What are the understory plant species for coffee? Hey, coffee is a nice big tall thing. So we could have things that maybe grew, 6 inches, 9 inches is our nice understory. How much work is it to have to go in and mow underneath all your trees? Is that not a pain in the butt? Don't do that anymore. Have plants that you never have to mow. How many of you go out into your fields and mow the weeds or till the weeds in? Stop doing that. Mother Nature, of course, bears soil. She will grow a plant in it. It's disturbed soil, so what's going to grow there? Weeds. So, I'm going to do a quick jump ahead. I have wonderful data. I can bore you with all kinds of data.
Tell them how to make compost and tea. I think you only have about 40 minutes left.
1 more minute. Okay, there's one more. I've got pictures from all over the world. From Battery Park City. So when the Twin Trade Towers collapsed, all of the property we were already working with downtown New York City was buried 10 to 30 feet deep in debris from the Twin Towers. So how do you recover all of those plants? Every single tree, 2,000 trees in Battery Park City were affected by that material coming down. How did we fix them? Ultimately, we have only lost 6 trees out of all of them that were affected. Compost peat is the way you get rid of salt problems. Golf courses, growing things in pure sand. How do you get the root systems growing down 5 to 6 inches in 3 months? Compost seed. Would you like the biggest pumpkins in the planet? Grown with compost. So getting the right biology into the system. So I can go on and on and on, but keep going. So lots of examples. There we go. Succession is what we want to come up on. Okay, way more examples. Roses, no diseases, no insect pests. We just got to get the right biology in there. Here's the food web that I was talking about.
That's the other poster, yeah.
That was the other picture that I tried to send so that you would have this. Sunlight plants, how far down do the roots go? Egg state, feeding bacteria and fungi. Oh wait, no root feeding nematodes. Exit them by getting the right biology in the system. Bacteria and fungi are eaten by their predators. Whenever their predators eat bacteria or fungi, we are releasing nutrients in the plants available for them, right in the root zone. That's why this works so well. We got to have higher level predators so we don't get unbalanced in the system. So now when we really want to take a look at what's the balance of the microorganisms, all this chemistry stuff. Isn't that nice to go right on by it? All that chemistry. Look at that compaction. We've covered pretty much all of that. I like this picture. Can't not show you. Pine seedling. This is the root system of the pine going down into the soil. And look at all that white fuzzy stuff on the root system. That's fungus. There's more fungal biomass on perennial trees when they're seedlings than there is seedling biomass. And it needs to have this kind of connection below ground. Every tree in the forest, every tree in your orchard, every banana plant in your plantation needs to be connected below ground to all of the other trees. All of those trees should be healthy together. They all share nutrients below ground. The fungus goes out into the soil and collects nutrients, brings them back to the plants. These mycorrhizal fungi, extremely important to get established. If you've ever used pesticides, they're not there. And you're gonna have to remediate that soil to get these mycorrhizal fungi to come back and reform these networks below ground for you. So, fungus. The plant is basically saying to the mycorrhizal fungus, hey, I need some phosphorus today. You go out and give me some phosphorus, and then I will give you some sugar. So the plant, the fungus says, no problem. I'm already getting phosphorus for myself. Let's just get a little bit more for you. So you will feed me some sugar. So, you know, it's kind of like the spider and the fly. Come into my root system, little mycorrhizal fungus. I got cakes and cookies for you. Mycorrhizal fungus says, cakes and cookies, I need cakes and cookies, so I'm going to grow into your root system. So the fungus gets in there, and the plant's feeding it, come into my parlor, little fly. And then the plant says, no more cakes and cookies for you. So you do something for me. And the mycorrhizal fungus goes, I need my cakes and cookies, whatever am I going to do? The plant says, I need phosphorus, nitrogen, sulfur, magnesium, calcium, and water. Fungus says, no problem. Already doing that for myself. Already have all of those enzyme systems functioning, doing that for myself. So I'll just get a little bit more, bring it in, feed the plant feeds the fungus, both go there. Want to get rid of weeds? Here's another component. Most of our weeds are non-mycorrhizal. And that's grass, not mycorrhizal. So get rid of some of the worst problems. I'm gonna get there. Soon. Here we go. Data. What is the proper balance for these organisms in the soil? As I said, we've looked at all kinds of soils from all over the world, all kinds of plant systems, in order to develop this kind of information for different kinds of plant species. Done a lot of this where we look at old growth systems, slightly less productive, not really very productive, and looked at the balance of fungi to bacteria. Lots of data on this to the point where they don't want to publish this anymore in scientific journals because it's opaque. So here's the summary of all the data, and you basically have a slightly different version of this. Early in succession, when Mount St. Helens went off, kaboom, instantaneously volatilized all life, poof, gone, in a cloud of CO2 because the temperatures were so high. The ash that falls back to the surface of the planet, sterile. No life at all. What comes back in first? Photosynthetic bacteria. Have to be photosynthetic because we don't have any organic matter in that soil. So the first thing that comes back in are the photosynthetic nitrogen-fixing, phosphate-solubilizing bacteria. These are the species of this group. They start putting out foods which allows true bacteria to start coming in, decomposers. Start growing more species of bacteria, keep up on that more and more and more as we go through the next couple months. Now we have enough bacteria so protozoa can come in and start cycling nutrients. Nitrite starts to be made. When we have enough nutrient cycling, phosphate solubilized, now fungi can come in, the other predators come into the system. Almost completely bacterial dominated. So lots of bacteria, very little anything else. Which fine, because at least we do have some nutrient cycling here. Allows weedy species. So the thistle, the nutgrass, the Johnson grass, the early successional weedy species, taproots getting punched down through compaction layers. Not very good structure in the soil. Fungal to bacterial biomass ratio, 100 times more bacterial than fungal. But all of these plants put in more fungal food, stalks, brown material, the cellulose, the lignin produced by any higher plant, more fungal foods than bacteria. And eventually we will shift the biology in the soil so we start growing early successional grass species, the bromas, the Bermuda grasses, brassicas like the mustard, cauliflower, the broccoli, things like that. Fungal to bacterial biomass ratio, still very much on the bacterial side. But they will, because there's more fungal food than bacterial being made, they will shift the species. We'll now have a little bit, just slightly on the bacterial side. We're growing mid-successional grasses and vegetables. Just keep going through the system. Our row crops are highly productive plant materials, grasses, equal biomass fungi to bacteria. The higher the biomass value of both of those, the more productive the system. So you want to increase production in whatever plant you're dealing with. You've got to maintain balance, but up the biomass of both of the type of organisms, as well as have protozoa and nematodes, microarthritis. Go over the edge into fungal-dominated systems. Shrubs and vines, 2 times more fungi than bacteria, to 5 times more fungi than bacteria. Putting more fungal foods in, woody stuff. So of course we're going to grow more fungi. Deciduous plants. So we're looking at bananas right in here. We're looking at coffee right in here. We're looking at shrubs and vines. So grapes and things like that. 5 times to 100 times more fungal than bacteria. Go into the conifer systems, our old growth forests, 100 times more fungi than bacteria, up to 1,000 times more fungi than bacteria. Mother Nature drives succession by ever increasing the fungal component in the system. What's the plant you want to grow? Well, where does it occur naturally? On the successional picture. Slightly different way to present it. The picture that you got as a handout. Slightly different version of what you got handed, but it's the same idea. I think the one you got handed is a little more easy to read.
Where would taro fit in a succession like you described?
Taro
What's kind of unique, yeah?
Well, let me go through this first, and then let's start asking questions about some specific plants. Barren soil, or where we only have some very disturbance-prone plants, very weedy species, 10 times more fungi than bacteria, almost no fungi to be found. If we start to grow weeds or brassicas, our ratio 100 micrograms of bacteria to 10 micrograms of fungi. Let's grow veggies. Most of our vegetables fall right in here. 500 micrograms of bacteria to 250 micrograms of fungi. Still nitrate-dominated bacteria, but we're getting a good component of ammonium, fungi. Now let's get more productive. Our highly productive grass system, our row crops, 600 and 600, really good production here. We have an equal amount of nitrate nitrogen as compared to ammonium nitrogen. Go over the edge into a fungal-dominated system. And this is where most of our coffee, most of our shrubs and bushes and things, deciduous trees, into the conifers, evergreen system. So taro, when it's in a riparian situation, when it's got wet feet, that soil is definitely going to be on the bacterial side, so you want it in this range somewhere. But as it dries out and it gets a little bit more mature, typically as we maybe go into the dryland part of the taro production, you want it to move into more equal biomass of fungi and bacteria. Rice is the same way. If we're going to allow a dry system where that soil dries out, it's not flooded anymore, we've got to go to the more fungal system to keep those plants alive and healthy. If they're in the riparian, the wetland system, then you need the fungi, but you don't need near as much. So those are observations working with some of the taro systems that I've been involved with. Lots more work needed there, though. More
You work in Hawai'i, taro?
Yeah, it works some, again, over on Maui, near Hana, the native taro production system there. Yeah, they've had their moments there, though, where things have been a problem. It had some diseases, and again, it's the imbalance of that biology in their ponded system.
So what is the published research that supports this? What cases?
So the ecological monograph is probably the best place to go, but when we're looking at publications, let me direct you to the journals that tend to publish this kind of information. And a couple of the names in this area. So David Coleman published a couple of books where we're looking at these kinds of ratios of fungi to bacteria. So there's a number of pubs in this area. He is one of the editors of Biology and Fertility of Soil. This is a journal. So a good journal to go and look in. Another good one from the Ecological Society, the Soil Ecology Society. That was the first president. Their journal that they publish is Applied Soil Ecology, and always has some papers in their issues that they come out. It's every 3 months. There'll be a new issue of that. Lots and lots of papers about biology and how we're shifting that ratio of fungi to bacteria. Andy Mozinki is another good person to look at his papers. He especially puts in the microarthropod part of the food web. He generally remembers, of course, most of my papers. Paul Hendricks. If you want a list of publications about compost tea, you can email Soil Food Web Oregon, and they will send you the list of pubs. Tom Pugh at Arizona State University has just done a whole reference list of these papers on compost tea, and as I recall, there's like 20 or 30 papers where it's all showing the disease suppressiveness of a good compost tea, where he's making it very clear that you have to look at the biology in your compost teas. There have been a number of academic people who have come to talks like this, and they hear me talk about compost tea. Let's make our compost with this mixture of high nitrogen, green, woody, make sure that it gets up to temperature, hot enough, long enough. We have really good biology in the compost. You take a small amount, like a pound of that compost, put it into a laundry bag or into a stiff bag where it will bubble air up through that, or just take your compost bag and swish it around in your 4 gallons of water for about 30 seconds. Mash it around, you know, just like you're washing a pair of dirty pants or something, but the compost instead, get that really well extracted into your water. We're getting all the organisms into that water. Pull the compost out, you should have all of the organisms in the water. A quick drop, look at it under your microscope so you know for sure. It's that easy to make compost tea. For a compost extract, add a little bit of food into that compost extract, let it aerate overnight, grow the microorganisms that you want to grow, compost tea. So that's the difference. An extract is just the extraction part of the compost, organisms into the water. Tea, we will add food. So we will grow the organism, fungal food or bacterial food. And most of the time it's fungi that we're lacking in soil. So really most everybody doing tea is making a fungal compost tea. So humic acid is the best material for growing fungi, but it has to be humic acid extracted from the compost. Do not go buy Leonardite extracted humic acid because it'll kill your fungi. The preservative that they use in Leonardite extracted humic acid will kill your fungi. So don't use that. Making a good compost, nice dark brown, rich color. How do you know you've got good humic acid in the compost that you made? Chocolate bar. Make sure that you got the color. And put your compost in like a sieve, a colander, pretty small holes in it, and just passively run, just pour water on the surface of that compost and let that water move passively through the compost, collect it when it comes out the other side. Nice dark brown color? Great. Not dark enough brown? Just take that water, run it through your compost again. Just keep running it through. until you get that nice dark brown color. If the water coming through your compost, you can't ever get it nice dark brown color, dump your compost out, get some more, run that same water through until you get enough humic acid into your water, nice dark brown 70% cocoa color, and there's going to be your good fungal food resource to get the fungi in your compost extract to grow. Now, if we're growing organisms in water, we better be applying some air bubbles. Better be aerating it, so we're not going to have a problem with going anaerobic in our tea. So, academic folks will come and listen to this talk. And what they'll do then is they'll go back to their place of business, and they say, for example, Dr. Linda Chalker-Scott at Washington State University came and listened to me, and she walked away saying, I'm going to prove that she doesn't know what she's talking about. She went home, and she grabbed manure. In what way is manure like compost? Not any color. Because manure is usually kind of a mix of straw and grains and wheat seeds and all kinds of stuff. What's the smell that comes from manure? It's anaerobic, isn't it? So she takes manure, puts it into the water, mixes the water around 5 minutes this way, 5 minutes that way, 5 minutes this way, 5 minutes that way. In what way is that like extraction? She takes that water, sprays it on her cherry tree, kills the cherry tree. Manure water. What's the biology in manure water? There's some pretty impressive anaerobic things in there, isn't there? A few cilia types of specs. So in what way would that biology have benefited that cherry tree? And yet you talk to Dr. Linda Chalker-Scott, and she says, I have data that proves that compost tea doesn't work. It'll kill your plant. No. She has data to prove that manure water that's anaerobic kills your plant. Nothing at all to do with compost tea, compost stuff. So most of the reports in the scientific literature that talk about compost kills plants or compost tea does not get rid of diseases are people who have never looked at the biology in either the compost or in the liquid stuff that they produce from that not compost. Because compost is, by definition, aerated mix of organic matter decomposed by bacteria, fungi, protozoa, nematodes, and microarthropods, then you better have all of those organisms in the compost, and you better extract them into the compost tea. No compost, it cannot be compost extract, it cannot be compost tea. It has to be showing the biological assessment. You read a scientific paper that in nowhere in that paper do they demonstrate an assessment of the biology using a microscope cannot fall for the fact that it didn't work. I'll give you everything every single time. Anaerobic, stinky, smelly stuff will kill your plant. But that's not compost.
Right now, you have an interesting situation in the island with coffee because one of the most effective ways to kill the coffee beetle borer is with a fungus, Bovarium bassiana. But right now, the grad student, the PhD student who has been working on the beetle borer is working as hard as he can to make sure that that we get approved to import bravado, which is like a chemical pesticide, a really powerful chemical pesticide. And the problem with Bavaria is that it's really expensive, so people wanted to grow their own, and that's not being approved, but they're trying to make bravado. It's like, what do you say to those people?
Bavaria bassiana is not very expensive if you realize that when somebody is selling that product, they want to tell you you have to use a whole quart-sized jug to make that compost tea. Absolutely not. Do not do that because you will fail. Way too much Bavaria in there. So when you're making, when you're using Bavaria, what you want to do is make a compost tea. So let's say you've got a 5-gallon bucket. You fill it up to 4 gallons of water. Aerate that water to blow off any of the chlorine or any of the chloramine. You've got to complex that with a couple of drops of the humic acid. I don't know if here on Hawaii your water treatment plants are putting chloramine in your water yet, or whether they're still just using chlorine. You might be one of the lucky places in the United States where they haven't started using chloramine. But over on Oahu, for example, in Honolulu, they add chloramine into the water because the chlorine isn't killing the pathogens anymore. And so we've got to put chloramine in there. Chloramine is not a gas. It does not blow off when you aerate. The only way to complex the chloramine is to put a couple drops of humic acid in there. So if you've got Leonardite extracted humic acid, fine. That works just fine for complexing the chloramine. Just make sure you put enough drops in that the water actually gets a slightly brown tinge to it. So now you've got enough humic acid to complex the chloramine, get rid of the thing that's going to kill your organism. What do we put chlorine and chloramine into our water for? Yes, and it works. So we've got to get rid of those things when we're making compost tea. So either aerate the chlorine out or add the humic acid to get rid of the chloramine. Now you have this slightly tinged bucket of compost, of water. We're going to take like a metal rod or something to just suspend a compost bag. So get a little bag where you can put about a pound of your compost in there. Make sure that it doesn't, that it doesn't twist. It's stiff. It won't twist around as you're aerating the water. So get like a 500-gallon aquarium aerator from the fish store. Take a couple of lines of Teflon tubing and just attach them to the bottom of your bucket. No aeration stones, no, bubblers, no nothing. Just the tubing. So that on the bottom of the bucket, you might have to get like a suction cup so you can hold the tubing, the end of that tubing on the bottom of your pail. So the air is going to come out and just bubble up. Should bubble right up to where your bag of compost is sitting. So as that air is coming up, it's hitting the compost, so the compost is churning and turning and mixing, and you're ripping the organisms off the compost into the water. Now, we want to add Bovaria into this system. Because each and every one of the spores of the Bovaria in that dried preparation that you bought, and this, like a quart of Bovaria costs like $500. You don't even think about adding that whole quart in there. You take a pinch. So grab your fingers, a pinch of your Bovaria spores, whatever sticks between your fingies, and you put that into your compost tea bucket. When we're looking at 4 gallons here, it's a pinch of the Bovaria spores. And you just put 500,000 Bovaria spores into that compost tea. During the brew cycle, so now we've got the compost, they're bubbling away, hitting the organisms, the humic acids, the fulvics, all those kinds of humic acid materials in the compost into the water. Let's add a little bit extra humic acid, so you've extracted your compost, pulled out some good humic, add that in there, maybe a little fish hydrolysate, a teaspoon or two. Fish hydrolysate, maybe a little kelp, half a teaspoon of kelp. And let that aerate overnight. Each one of those Bovaria spores will germinate. They will start to grow. So when you spray that out, the instant it hits one of those beetles, it attacks, it consumes. It's already activated. It's already ready. It is looking for that beetle. It's looking for some disgusting little insect to go out there and eat its way through their surface, their exoskeleton, get inside and turn that whole aphid, whitefly, cutworm, root grub, beetle, whatever you want, take care of your problem. So here's a quart of Bovaria spores that you bought for $500. How long is this going to last you? the rest of your life. Share it with your friends, because, that's more than you possibly needed. See why the people who sell Bavaria don't want to tell you about what I just told you? Because how are they going to make any money? So they want you to dump the whole quart. Take your Bavaria spores, put it into water, spray it out with the water. Well, what's going to activate your Bavaria spore to germinate and grow? Nothing. So they're usually not even successful if you do what they talk about. So put it into your compost tea, man, that stuff works like magic. So we have these beetle infestations. Japanese beetle on the east coast of the United States. We just wipe out most of the Japanese beetles. So really, think about this. Do you want to use Bavaria as a preventative? You're going to kill any insect that it lands on. How about your green lacewings? How about your ladybugs? How about your... It'll take out banana weevil, no problem. Especially get the juvenile stages in the soil. So make sure you put it not only on the above ground part where the adults are, but make sure you apply it into the soil. But then you might have to come back in and use a compost to reapply, to reinoculate your really beneficial microarthropods back into the system. So you only use Bavaria, you only add that into your compost tea when you have an outbreak of a problem insect. And that problem insect is about to take out your coffee, or it's about to take out your broccoli, or it's about to take out your whatever plant. You use it as a pesticide, because isn't that what it is? It's a biological pesticide. Do not use it as a routine, because would we not like to have honeybees still left in our lives? It'll take out your honeybees. So don't be using it except when you have to. So Bavaria, trichoderma is another one. You got a fungal outbreak happening, you got phytophthora, you got mildew, you got blight, you got something else wiping out whatever plant you're trying to grow. Buy some of the trichoderma. Again, it's going to cost you maybe $100 for a container of trichoderma, but it's all spores in there. In a 4-gallon compost tea, 4 gallons will cover 4 acres. So on these sorts of things, you may only have to put a gallon. How many of you can apply compost tea 1 gallon per acre evenly out over the whole acre? Not happening, is it? So you're going to take those 4 gallons and you're going to put it into however much water you need to put it into to get it sprayed out. What if I only have an acre? I just made 4 gallons of this compost tea. Go ahead, put it out loud on your 1 acre. More the merrier. Doesn't, but you're going to have to at least put a gallon per acre, and it's going to have to be pretty good stuff. Trichoderma then, pinch your trichoderma. This is a couple million spores between my fingers. Put that into the compost tea. Overnight, the trichoderma will germinate and grow on the food resources coming out of the compost. So they are all alive, active. You spray that out onto the place where you got the fungal disease happening overnight. You're going to take care of that mildew or the blight or the fusarium or the, botrytis, whatever you got. But again, trichoderma, you put that out, you're taking out all fungi. They are not specific. So you use a trichoderma to get rid of disease that's wiping you out, okay. But now you're going to have to make another compost tea with as much really good fungi as you possibly can. You're going to have to come in and apply that. After about 7 to 10 days, once the trichoderma has wiped out all your fungi, then you better reinoculate, because that trichoderma is a pesticide. Applied in that fashion, trichoderma can really wipe it out. It can wipe out all the mycorrhizal fungi in the roots. Ooh, so careful. These things are not to be used routinely. They're used when you've got a problem. When you don't have the right biology in your soil, and yes, it's a last ditch, we got to save your plants, save your plants. But then let's come back in and put biology in so you don't ever have to do that again.
I want to ask a question about, I'm going to use a specific plant, but I want you to answer it by using the concept so that it can be used for other instances. If I follow your succession right, I grow tea, so I should have a slightly fungal dominant,
And think about where along the
shrub brush area, but they're supposed to be growing in really, really active soil, 4 to 5, which would be bacterial. So I'm having trouble wrapping my...
4 to 5 would actually be anaerobic bacterial. The only way to have a soil pH of 4 to 5 would be, you're going to have really sick plants. So let's go back and see who's giving you the recommendation of a pH of 4 to 5 in those root systems.
Everybody who's grown tea in the last 5,000 years.
Yeah.
So do I ignore pH?
You let the plant take care of pH all on its own. Because what we're seeing, like in Japan, where we've been working with tea production systems, or South Africa, where we've been working with tea, southeastern part of the United States on tea, is that the most productive tea plants are not growing in the soil down around 4 to 5 pH. They're growing in a highly fungal dominated soil. And the pH of that soil is between 5.5 and 6.
So
there's some real mess up that's happened.
Succession chart rather than the pH recommendation.
Right. The plant will make the pH what the plant wants it to be. By controlling, is this gonna, am I gonna feed more fungal food out there, or am I gonna put more bacteria? So and when you start really looking at that root system, what you discover is that right here, I'm extremely bacterial, because what the plant needs is what feeds bacteria. But over here, I'm really fungal. There aren't any bacteria to be found. All fungal. And over here is, equal biomass. Over here is a little bit fungal. Gosh, by the time you start summing over that whole root system, you can come up with any answers you want. See how, isn't that joyful when you really start understanding biology? That's the plant being in control. Stop pretending like we know. What do we know? Nothing. Okay, maybe a little. We have a lot yet to learn, and in large respect, it's just maximize diversity. Let the plant do what it's going to do. Make sure it doesn't go anaerobic. Make sure we got the biology that's building structure. Those root systems just are not as deep as they possibly can. Every year be documenting the root systems are going deeper and deeper, and then you know you're doing the right thing. So you really have to be mentioning biology in your soil all the time. If you've got a little patience and you can be, we're just watching and observing, you don't really have to do a lot of measurement. But a lot of people are doing this for a living, and it's the only way they're getting money. Really pays to go ahead and look at the biology before really to speed this whole process up.
Can you point us to resources for creating this compost? Best practical procedures.
Well, let me just.
Go to the Soil Food Web website, so soilfoodweb.com. Soil. It is amazingly enough my website. And there's a book on there. So soilfoodweb.com. And when you go to the first page, so our home page, there's a set of tabs up here. And you want to go to click on the tab that says About Us. That's a tab you want to click on. It takes you to a page that tells you how to sample soil, how to send your soil into a blah, blah, blah. But also on that page, there's a series of tabs, and you want to click on the one that says SFI Approach. This is the book. So when you click on that page, what comes up is a table of contents. Down this side is a table of contents. And it's got like compost, it's got compost extract, it's got compost cheese, it's got the food web. So go to the section on compost if what you want to find out about is compost. Or go to the section on compost tea. If you go to the Rodale website, most of the books, so you can buy copies of these that are a little bit more expanded. I have a book on, well, I think we just took the book on compost off because we're revamping it from the data that we've been collecting at Rodale over this last year. So we're going to have putting on a troubleshooting page where what happens when I've got lots of wet spots in my compost? What can I do? What happens when my compost gets real dry? What do I do? What happens when I can't generate any temperature at all? What do I do? So that troubleshooting page is we're adding that into the compost book, so that will be re-released from Rodale. The compost tea, probably the book that really covers a lot of this in pretty simple language. So I realize that it's come to my attention over the last several years that I talk like a scientist. And maybe not in the easiest to understand language all the time. So we just published a book called 10 Steps to Gardening with Nature. So small scale, so we're really dealing with systems that are going to be like 5 acres or smaller, backyard systems. And recipes for the compost teas, a lot of that information is in that book. So, and it's really relatively inexpensive. I think it's like $35, $40, something like that for that one. Give them to all your friends for Christmas.
We have 2 more minutes.
Okay, so there's some of the documentation. Have fun going to the Rodale website and looking at all my books. Buy them all. That was out and somebody trying to sell you something. Yeah.
I know this isn't soil, but do you know any effective methods of dealing with floods that have a bad disease carried by the floods?
Floods are attracted by anaerobic conditions. So the only reason you've got flood problems is because there's some anaerobic smell that's being produced in your soil or in your organic matter layer. That's what attracts floods. So you've got to do something to get rid of the compaction that's down there. Now your soil may seem nice and light and fluffy up here on the top, but start poking your fingers down through. I was dealing with the natural agriculture people in Japan. And so they had nice fluffy soil on the top of the soil, and I said, so how'd you achieve that? And they said, well, we till the soil so it makes it nice and fluffy. Right.